everyone and welcome to Miss Estruch Biology. This video is going to be a mega one and so helpful for you to prepare for your exams because I'm going through everything you need to know for AQA A-Level Biology Paper 1. That means it's going to be long though, so you can use ×2 speed to get through it quicker or skip to the time codes for the different topics that you need. And if you do need any more extra help, then check out the resources I have in my description that are there to boost your grades, such as my A-Level notes or my flashcards. But for now, let's get into it. So monomers are smaller units which can create larger molecules, and the polymers are made from lots of monomers which are bonded together. And the examples that you need to know are glucose, amino acids and nucleotides, so RNA or DNA nucleotides for the monomers, and then the polymers which those monomers can create. So glucose can create starch, cellulose, glycogen, amino acids can make proteins, DNA and RNA polymers. Now to create these polymers, it is a condensation reaction. And this would be a 2 or 3 mark definition. So joining two molecules together would be the first mark, creating a chemical bond would be the second, and removing water would be the third. To hydrolyze, which means to break apart or to split the monomers, that would be a hydrolysis reaction. And it's still 3 marks, but the opposite 3. The breaking of a chemical bond between two molecules and involves the use of water. So carbohydrates is the first biological molecule that you need to know. And I've got an overview here of the 3 levels of size of the molecules you need to know. So monosaccharide, mono means 1, so 1 sugar unit. Disaccharide, di is 2, so that's when you have 2 sugar units joined together. And the polysaccharides is when you have many joined together. And for each of those carbohydrates, there are 3 examples that you need to know. Glucose, fructose and galactose are the 3 monosaccharides that you need to know. The disaccharides are sucrose, maltose and lactose, and the polysaccharides, starch, cellulose and glycogen. Now for the monosaccharides, the main thing you need to know is the structure of alpha and beta glucose. So alpha glucose we can see here. This is the level of detail that you'd be expected to draw this for AQA biology. And you would also need to know the formula, so C6H12O6. Now I did say you need to know alpha and beta, and that's because glucose comes as 2 isomers, which is when you have the same molecular formula, but there is a different structure. And the key differences I'll just highlight here. So for alpha glucose, on the carbon 1, which is the carbon that would be in this position, you have the hydrogen atom on top and the hydroxyl group on the bottom. For beta glucose, the only difference is those swap around. So the hydroxyl group's on top and the hydrogen atom is on the bottom. The disaccharides then is made up of 2 monosaccharides. When those are joined together, the chemical bond that forms is a glycosidic bond, and they are created via a condensation reaction. Now I did name the 3 that you need to know, but in addition to that, for those 3, you would need to know the word equation and therefore which 2 monosaccharides they're made from. So maltose is glucose plus glucose, lactose is glucose plus galactose, easier one to remember because there's lactose in the name, and then sucrose is made up of glucose plus fructose. And because all 3 are condensation reactions, that is why one of the products is water, because water is released. Now for the polysaccharides, you need to know the structure and how that links to the function, as well as some general other facts. So here's a very basic summary. Starch and cellulose are both found in plants, but they have different functions. Starch is a store of glucose, so it can provide chemical energy. And the cellulose is also in plants, but the function is structural strength in the cell wall. Glycogen is the only one found in animals, and this is a store of glucose as well, mainly found in the liver and the muscle cells. So just to summarize everything you need to know about the carbohydrates polymer, which is the polysaccharides, you need to know which monomer they're made of. Now, yes, it is glucose for all 3, but which isomer is different. Starch and glycogen are both made from alpha glucose, but cellulose is beta glucose. They're all glycosidic bonds, but they're different types. And the thing that's different about them is the location. And that's what these numbers refer to. So a 1 to 4 glycosidic bond means the bond forms between carbon 1 in one of the molecules and carbon 4 in another. And those numbers just refer to the position of the carbon in the glucose ring. So starch is made up of 1 to 4 and 1 to 6. And amylose, which is one of the polysaccharides of starch, only has 1 to 4, whereas amylopectin has both. Cellulose only has 1 to 4, glycogen also has both. And it's the 1 to 6 glycosidic bonds that create a branched structure. The 1 to 4 forms polymers in a straight line. The function we've already said on the previous slide, as have we said the location, but a little bit more about the structure then. And this again links to what we were just saying about the bonds. So amylose is an unbranched polymer, and it actually coils up to make a helix. And that is really useful because if it coils up, it can then be compacted to fit a lot in a small space. Amylopectin is branched, and the advantage of that is the branches create a larger surface area, so more enzymes can attach to the end and hydrolyze to turn it back into glucose when the plant might need glucose. All three have one feature in common. They're polysaccharides, which means they're large. And because they are large, they're insoluble. That means they won't affect the water potential of the cell and therefore no impact on osmosis. Cellulose has got a very different structure, and this is because it only contains 1 to 4 glycosidic bonds. So the polymer forms long straight chains. Now those chains line up in parallel next to each other and hydrogen bonds join them together. And we call that structure a fibril. Because there are so many hydrogen bonds holding these chains together, collectively they provide a lot of strength. And that is why cellulose is a very strong molecule. It's the large quantity of hydrogen bonds. Glycogen, now this is actually very similar to starch, in particular the amylopectin in starch. The key difference is it has a higher proportion of 1 to 6 glycosidic bonds, and for that reason it's even more branched and it can be even more readily hydrolyzed back into glucose. And that's an advantage because we find in animals, and because animals move, they will need more glucose. The next molecule is the lipids, and there's two lipids you need to know, triglycerides and phospholipids. So here is the level of detail you need to know their structure. They both have a glycerol molecule, and for the triglyceride, there are 3 fatty acid chains that come off that. So tri for the 3, tri meaning 3. Phospholipids, the key difference is one of those fatty acid chains is lost and instead it has a phosphate group attached to the glycerol molecule. Now how they form is the same, but I'm actually just going to go through it with the triglycerides. So it is a condensation reaction, and it will be 3 condensation reactions because there are 3 fatty acids that are going to be joining to the glycerol. Now, although it's a condensation reaction, that does not mean this is a polymer, and it isn't a polymer because it's not many repeated units joining together. So what we have is 1 water molecule is lost between each of the fatty acids and the part of the glycerol it's attaching to. So in total, that means we have 3 water molecules lost and 3 condensation reactions occurred. And the bond that forms is called an ester bond, and we have 3 ester bonds. So that is the structure of a triglyceride. A little bit more about the structure is the fatty acids. You do need to know that they can be either saturated or unsaturated. And a saturated fatty acid means that there are no double bonds between any of these carbon atoms. So you only have single bonds between the carbon atoms, and therefore it's fully saturated, holding the maximum amount of hydrogen. But this here would be your 1-mark definition in an exam. Unsaturated fatty acids have at least 1 double bond between the carbon atoms, but you do have to state between the carbon atoms in the exam to get the mark. The properties then of the triglycerides. So the function is as an energy store. And the reason it stores lots of energy is because of the large ratio of the energy storing carbon to hydrogen bonds. And that is compared to the number of carbon atoms. So you have lots of energy stored in those bonds. The second reason is due to the high ratio of hydrogen to oxygen atoms, and that can actually act as a metabolic water source. So triglycerides can release water if they are oxidized. And that's actually what camels have in their hump. They have lipids, triglycerides, they don't have water. Triglycerides also have a similar property to the polysaccharides in that they do not affect the water potential and therefore affect osmosis. And this is because they are large and also hydrophobic, which means they will repel water. The last property is lipids have a very low mass compared to other types of tissue in your body, for example, muscles. And that's an advantage because it means you can store a lot without increasing the mass as much as extra muscle would. So you can store a lot of this for an energy storage without increasing the mass as much as other tissues would. Next then, the phospholipids. So we've already talked through the difference in the structure. It's still made by condensation reactions, but it would only be 2 condensation reactions because there's only 2 fatty acid chains. Still an ester bond, but we'd only have 2 ester bonds. And we can see up here, that is where the phosphate group attaches to the glycerol. Now this phosphate group is what gives the phospholipids some very different properties to the triglycerides. The phosphate group has a negative charge, and due to that negative charge, that bit, so the hydrophilic head, will actually attract water, but it will repel lipids. The fatty acid chain, so the hydrophobic tails, they don't have any charges on them. So they're actually described as hydrophobic, meaning it repels water, but they are able to mix with other fats or other lipids. And that is why the phospholipids can form this bilayer. We have these 2 charged regions, and in water, these phospholipids would position themselves so that the heads are on the outside, exposed to the water because they are hydrophilic, meaning they're attracted to the water. But because the tails are hydrophobic, meaning they are repelled or they repel water, they would spin round and the tails would face each other so they're not in contact with the water. Proteins then is our next biological molecule, and they are another example of polymers, and the amino acids are the monomers that they're made up from. You do need to know how to draw this general structure of an amino acid, so it is one of the things you could be assessed on in the exam. Now, a way to help you to remember it is to box it into these key groups. You have a central carbon in the middle of the molecule. There's a hydrogen atom that comes off, and an R group that comes off the top. Now, those could actually be either way round, top or bottom. The R group represents the variable group, so that changes for all 20 different amino acids. The amine group or amino group, that will always be present, and that is NH2, and the carboxyl group, that will also always be present, C double bond O, OH. Now, to make a dipeptide, which means 2 amino acids bonded together, it would be a condensation reaction, so water would be removed. The bonds that would form would be a peptide bond. To make a polypeptide, that would be when you'd have multiple amino acids joined together and multiple condensation reactions, still all joined together by peptide bonds. So that would create your primary structure of a protein, but that primary structure gets modified into the secondary, that gets modified into the tertiary, or it could be a quaternary. So we're going to go through what all of these 4 levels of organisation or development of a protein look like and how they're held in place. So the first level is the primary structure, And this is what is made straight after translation in protein synthesis. And the definition for this would be a 1-mark question. It's the order, or you could say the sequence, of amino acids in a polypeptide chain. So that's your polymer. The secondary structure then is when that primary structure is folded, or could be modified by twisting, because we can see here the alpha helix. But that would be the key marking point, that we then have an alpha helix or a beta pleated sheet that's created, and those are held in place by hydrogen bonds. The secondary structure then gets modified further, so it's further folded to create a unique 3D shape. And that shape is held in place by ionic, hydrogen, and sometimes disulfide bonds. And it's actually the primary structure that determines the location of these bonds, the ionic, hydrogen, and disulfide bonds. And it's the location of the bonds which determine how it folds and the 3D shape. The final level of organisation is the quaternary structure. That's cut off slightly, but that says structure there. Now that is still this unique 3D shape with the same bonds, but the only difference is it's a protein that is made up of more than one polypeptide chain. But it is still basically the tertiary structure. You just have more than one chain, so we call it quaternary. Enzymes are an example of proteins that you need to know. So an enzyme is a protein in the tertiary structure, so that unique 3D shape, and their function is that they catalyze reactions, and they do this by lowering the activation energy of a reaction. Now every enzyme is specific, and what that means is it can only catalyze one particular reaction, and that is due to the unique shape of the active site, which, this is an application of what we just said in the slide before, that primary structure determines the locations of the bonds, that determines the folding and the unique shape. So this is why each enzyme can only catalyze one particular reaction, and you get that unique or specific active site. So in that way, the active site is complementary in shape to a particular substrate. Now there's actually different models which explain how enzymes work. And at GCSE you would have learnt the lock and key model, but the accepted model currently is the induced fit model. So that is what you'd be expected to talk about at A level. You wouldn't be expected to mention the lock and key method. So the induced fit model is one that states that the enzyme's active site is induced, or it slightly changes shape to mold around the substrate. So initially, the substrate and active site are not completely complementary, but as the substrate binds, that causes the enzyme's active site to slightly change shape and mold around. That moving around the substrate puts strain and tension on the bonds, and therefore less energy is needed to break the bonds. And that is how enzymes lower the activation energy, which is the amount of energy needed for a reaction to occur. There are 5 factors that you need to know that affect the rate of an enzyme-controlled reaction: temperature, pH, substrate concentration, enzyme concentration, and inhibitors. So let's have a look at each one. So for temperature, if there is a lower temperature, that would mean that the molecule, so the enzyme and the substrate, would have less kinetic energy. Therefore, they won't have as many successful collisions, and you'd have fewer enzyme-substrate complexes. That is why the rate is lower at colder temperatures. Above the optimum, though, there is now so much kinetic energy that it causes some of the bonds to break. So, for example, the hydrogen bonds might break, and that means that the protein loses its unique 3D shape. The active sites change shape, and therefore you won't have enzyme-substrate complexes forming, and the rate decreases. For pH, either side of the optimum pH, which actually can vary depending on where the enzyme is found, either side we have a very rapid denaturing of the enzyme. And that's because either too high or too low a pH will interfere with the charges in the amino acids found at the active sites. That can cause the hydrogen and the ionic bonds to break, and again, the loss of that tertiary structure and the active site changes shape. So we describe that as the enzyme denaturing, and again, there'd be fewer enzyme substrate complexes, and therefore the rate of reaction decreases. Substrate and enzyme concentration have a similar idea behind them. There's no enzyme denaturing, but if we have a look at this one first, if there's insufficient substrate, there will be fewer collisions between the substrate and the enzymes, and that's why the rate of reaction is lower. But if you add more and more substrate, but no extra enzyme, eventually you'll get to the point where the enzyme active sites are all in use or they're saturated. So even if you add more substrate, there's no more free enzymes, so the reaction can't go any faster. So the rate remains constant. For the enzyme concentration, if there's insufficient enzymes, so at these low concentrations, then the active sites will become saturated with whatever substrate is there. And that's why if we don't add more enzyme, the rate will stay low, but as you add more enzyme, the rate will increase. However, you'll get to a point though where if you keep adding more and more enzymes but don't add any more substrates, you'll just have a surplus of enzymes and there isn't any extra substrate for those enzymes to bind to, so the rate won't increase any further. The last one was the enzyme inhibitors. Now both type of inhibitor, the competitive and the non-competitive, both bind to an enzyme. So in the exam, you have to be specific and say which part of an enzyme they attach to get the mark. A competitive inhibitor binds to the active site, so we can see that here. And if it can bind to the active site, that means this inhibitor must be the same shape or very similar in shape to the substrate. And if the inhibitor is bound, that will prevent enzyme substrate complexes forming. So if you add more substrate, for a competitive inhibitor, the substrate will actually eventually be able to knock out the inhibitor, take its place, and therefore with a very, very high concentration of substrate, the effect of the inhibitor is no longer seen. However, a non-competitive inhibitor binds to the allosteric site, and that is a part of the enzyme away from the active site. So it doesn't bind to the active site. But as it binds, it causes the active site to change shape. And for that reason, the inhibitor has made it impossible for enzyme substrate complexes to occur because the substrate is no longer complementary to that active site and it can't bind. So that is how these inhibitors lower the rate of reaction. And even if you add more substrate, that won't help because the active site's a different shape. Now you could be asked for the biochemical test for all of those molecules that I've just gone through, or some of them anyway, the ones that I'm going to go through. So the first one is the biochemical test for starch. This would be a 2-mark question to describe the method. You add iodine and a positive test result would be the iodine goes from orangey brown to blue black. The test for reducing sugars, you would add Benedict's reagent and you have to heat it. So you need both of those ideas to get the mark. The positive test result would be the original blue colour would turn either green, yellow, orange or brick red. And those different colours indicate the concentration of reducing sugar present. So the more red it is, the higher the concentration of reducing sugar. The test for a non-reducing sugar test then, you would first of all have to do your Benedict's test for reducing sugars, and if it remains blue, which means a negative test result, you then go on to these stages. You would add acid and boil. You have to say boil because it does need to be above or at 100 for this to work. You would then cool and neutralise the solution. Because you've cooled it, you would then have to say heat and add Benedict's reagent again. And this time the positive result would be the solution goes from blue to either orange or brick red. Now it would always be orange or brick red this time because if it was a non-reducing sugar, that means it was probably sucrose. and sucrose is made up of glucose and fructose. So when you hydrolyze it, that means it goes to two sugars, so glucose and fructose, instead of just one sugar, sucrose. So you've now doubled the concentration of sugar present, so it will always be orange or brick red. The test for proteins is you add biuret, which is blue in colour, and if you have a protein present, it will go purple. For lipids, it would be a 3-mark question. You have to dissolve your sample in ethanol, and you would do this by adding ethanol and shaking. And sometimes there is a mark for saying you have to shake. Once you've done that, you then add distilled water, and a positive test result would be white in colour, and we describe it as an emulsion, this sort of thick, milky texture. So the next biological molecule is DNA, or deoxyribonucleic acid. And this function is it codes for the sequence of amino acids in the primary structure. So it's very, very essential. It contains the genetic code and it can be passed on to make new cells, but also it can be passed on to the next generation. Now, DNA is a polymer, and you get two polymer chains joining together to create a double helix. So the monomer, you would have to be able to draw this in this level of detail. We have our phosphate group attached to a pentose sugar and the nitrogenous base. But because this is a DNA nucleotide, you would have to say that the pentose sugar is deoxyribose, and the nitrogenous bases, or nitrogen-containing bases, are guanine, cytosine, adenine, and thymine. So to make the polynucleotide, which is the name for the polymer, it would be a series of condensation reactions where water is removed and a phosphodiester bond forms between the deoxyribose and the phosphate group of another nucleotide. And that makes this sugar phosphate backbone, which is very, very strong because a phosphodiester bond is a strong covalent bond. Between the two polymer chains, we get hydrogen bonds, and those form between the complementary base pairs, which are cytosine and guanine, and adenine and thymine. RNA is the other nucleic acid that you need to know about, and it's almost identical in shape or in structure. The key differences are that instead of having deoxyribose as the pentose sugar, it has ribose. It doesn't have the nitrogen-containing base thymine, it has uracil. Now, RNA is also, in terms of the polymer, it's much shorter than the DNA polymer, and that's because mRNA is only a copy of one gene, whereas DNA is all of the genes, and also tRNA is relatively short also. And all of the polymers are single-stranded for RNA. Now, the functions you actually learn in more detail in topic 4, but you do need to know about one particular RNA molecule called rRNA. That combines with proteins to make ribosomes. So DNA replication then, in order for new cells to be created, every new cell needs their own copy of the entire genome, which is all of the DNA. So the DNA must replicate before a new cell can be created. And the way DNA replicates is described as semi-conservative replication. And what that means is one of the original strands of DNA combines with one newly synthesized strand to create the new molecule. So how that happens? Step 1, the enzyme DNA helicase breaks hydrogen bonds between the complementary base pairs. And that causes the double helix to unwind and the two strands to separate. And both of those strands will act as a template. Free-floating nucleotides within the nucleus will then align opposite the complementary base pairs, and the enzyme DNA polymerase will join adjacent nucleotides together. So it's joining the nucleotides together to make phosphodiester bonds. So now we have our newly synthesized DNA combined with the template strand, and that is our new molecule of DNA, or the daughter DNA. The evidence for this you need to know a little bit about also. So it was Watson and Crick who discovered the structure of DNA, so that double helix, but they only managed to discover that because of Rosalind Franklin's research on X-ray diffraction. Michelson and Stile, they conducted experiments which proved that DNA replication was semi-conservative. Now, ATP is what we call a nucleotide derivative, and that's because it's very similar in structure to DNA and RNA. It still has a pentose sugar, it still has a nitrogen-containing base and a phosphate group, but it actually has 3 phosphate groups. It will always have ribose and it will always have adenine. So adenosine triphosphate is what ATP stands for. And its function is it's an immediate source of energy for biological processes. So in other words, it's used in metabolism, which is all of the chemical reactions within a cell. ATP is made during respiration by ADP and PI, which is inorganic phosphate, joining together in a condensation reaction using the enzyme ATP synthase. Now, ATP releases energy when it is hydrolyzed. So when the bonds between the phosphates are broken, and we then have ADP plus that inorganic phosphate, that releases energy. And the enzyme ATP hydrolase is what catalyzes that reaction. Now, ATP can also do something called phosphorylation. And that is when that inorganic phosphate is actually transferred and bonds to a different compound. And in doing that, the compound that it binds to or bonds to becomes more reactive. And that happens to glucose at the start of respiration in glycolysis. The next biological molecule is water, and water has 5 key properties that you need to know about. Now, water is very, very important because it makes up about 60 to 70% of your body. And the reason it's so important links to these properties. Now, most of the properties are due to the hydrogen bonds which form between water molecules. And those will form between the oxygen of one water molecule and the hydrogen in another. Water is also described as being polar or dipolar because the oxygen has a slight negative charge and the hydrogen has a slight positive charge in the water molecule. So the 5 properties: number 1, water is a metabolite, which means it's involved in chemical reactions. So we've already seen in this video how it's involved in condensation and hydrolysis reactions, but it's also involved in photosynthesis. Because of its dipolar nature, it's a very, very good solvent, and this is important because if it can dissolve solutes, then they can be easily transported around the body in the cytoplasm in a cell, or in the plasma of blood, or in the liquids in the phloem and the xylem in plants. Chemical reactions also happen more readily in liquids. It has a high heat capacity, which means it takes a lot of energy to raise the temperature. And in that way, it can buffer temperatures. So that means the fact that our body is mainly water, it takes a lot of energy to increase our body temperature. And that's good because we don't want enzymes to be denaturing. It also has a large latent heat of vaporization, and that provides a cooling effect because this property means a lot of energy is required to convert water from its liquid state into its gaseous state. So if water does evaporate, that means a lot of energy has been transferred in that process, and it provides a cooling effect when we sweat or when plants are going through transpiration with water evaporation. Water also has strong cohesion, and this is due to the hydrogen bonds between the different water molecules. And cohesion means the water molecules are sticking together. And in plants, this is particularly important because that means you get this continuous column of water moving up the xylem. It is also what provides surface tension to water, and that can actually provide a habitat for certain organisms. Inorganic ions is the last part of the specification for this topic, And what you need to know is that ions can occur in solution, so for example in the cytoplasm of cells, in other bodily fluids as well like the blood, sometimes in high concentration, sometimes in low. And there are a selection of inorganic ions that you need to know and what function they have. Now this actually links to other parts of the spec, but the hydrogen ions, you could look at the importance of them in terms of how they can alter the pH of a solution, and that could have an impact on enzymes, but also hemoglobin, if you think about the Bohr effect that comes up in another topic. Or you could look at the importance of hydrogen ions in chemiosmosis, in respiration and photosynthesis. Iron ions are a component of hemoglobin and are involved in the transport of oxygen. Sodium ions are involved in the co-transport of glucose and amino acids in absorption. Or you could look at the role of sodium ions in action potentials as well. Phosphate ions are found in DNA, RNA and ATP. The importance in DNA and RNA is the phosphate group is where the phosphodiester bond forms. In ATP, the phosphate group can be added to other compounds to make them more reactive. So within the eukaryotic cells, the key organelles that you need to know the structure and function of are all the ones listed here. And we're going to go through each of them one at a time. The nucleus, the key structures within that are the nuclear envelope, nuclear pores, nucleoplasm, chromosomes and the nucleolus. And the overall function of the nucleus is where DNA replication occurs, and it's a site of transcription, which is where mRNA is made, and it contains the DNA, which is the genetic code for the cell. We also have within the nucleolus, that is the site of RNA production, and it's where ribosomes are made. The endoplasmic reticulum, you have the smooth and the rough. And we can see here the top diagram, we've got the rough endoplasmic reticulum, and we can tell because we've got the ribosomes on the outside. And the other side of this top diagram is the smooth endoplasmic reticulum, and that has no ribosomes on the outside. Now the RER, which is the rough endoplasmic reticulum, that is where protein synthesis occurs because of those ribosomes on the outside. The SER, the smooth endoplasmic reticulum, that is where you have the synthesis of lipids and carbohydrates, and they also get stored there as well. The Golgi apparatus is a folded membrane making this cystine shape, and we have vesicles that will pinch off from those cystinae once whatever it is that's going to be modified has been modified and packaged. And we've got the whole list here of the different things that would happen. So it could be that the carbohydrates are being added to proteins to make glycoproteins, which might be embedded within the cell membrane. You can also produce secretory enzymes, carbohydrates, transport and modify lipids. We get lysosomes. Molecules are also labelled with their destination, essentially meaning they're going to have a molecule added so they'll be able to bind to receptors on the target cell for where that molecule is needed. Lysosomes are bags of digestive enzymes, and this is often involved in phagocytosis. So the enzymes that are required to hydrolyse the bacteria or virus, whatever it might be, would be in a lysosome. There'll be exocytosis, which is where the products are going to be released to the outside after it's been destroyed. The mitochondria is the site of aerobic respiration. So you'll have lots of ATP production here. It's a double membrane organelle with the outer membrane, and the inner membrane is this folded part making up the cristae. And the inner membrane is where oxidative phosphorylation happens, which is one of the key stages in aerobic respiration. They have their own loop of DNA, which is very similar to prokaryotic DNA. And this is so that they can code for the enzymes that they need in respiration. Ribosomes, now these are found in prokaryotic and eukaryotic cells, and that is because they are not membrane bound. Ribosomes are just made up of rRNA and proteins. You do have different sizes though, so the ATS is the larger size ribosome in eukaryotic cells, 70S is the smaller sized ribosome found in prokaryotic cells, as well as in mitochondria and chloroplasts so that they can make their own enzymes. And it's the site of protein synthesis. The vacuole, which is only found in the plant cells, not in animal cells, this structure is filled with fluid and surrounded by a single membrane called a tonoplast, and this is what helps to make the cell turgid and provide support. You'll have temporary stores of sugars and amino acids within the fluid as well, and the pigments within it may colour the petals, and that's what helps to attract pollinators. Chloroplasts, again, only found in plants, and this is the site of photosynthesis. You have, again, it's a double membrane organelle, and we have the outer membrane, the inner membrane, and then on the inside we have even more membranes, and that is these thylakoid membranes which stack up to create what we call a grana if it's plural, or just a granum if we're just looking at one of those stacks. And the thylakoid membranes are embedded with proteins and pigments such as chlorophyll, and therefore it's the site of the light-dependent reactions in photosynthesis. The stroma, which is the fluid part surrounding the thylakoid membranes, you have lots of enzymes in this location which are needed for the light-independent stages of photosynthesis. Cell walls are not in animal cells, but they are in fungi and plant cells, and they help to provide structural strength to the cells and prevent them bursting if lots of water moves in by osmosis. And that's because they contain a particular molecule that provides strength, and in plants that's cellulose, and in fungi that would be chitin. The plasma membrane is found in all cells, and this is in the cell membrane, so the cell surface membrane, but also when we say it's a double bound or membrane bound organelle, it's also made up of this phospholipid bilayer with the different molecules embedded within it. And the function of the plasma membrane is it controls what can enter and exit the cell. Prokaryotic cells, for example bacteria, you need to be aware of the key differences. So it could come up as a comparison or a contrast question. So first of all, the cells are much, much smaller. They don't have any membrane-bound organelles. They do have ribosomes though, because ribosomes are not membrane-bound, but they have the 70S size ribosomes. They don't have a nucleus because that is a membrane-bound organelle. So instead, their DNA will just be as a single loop, which is free within the cytoplasm. They do have a cell wall, and that is made up of murin. Sometimes they might also contain plasmids, which are additional loops of DNA. They might have a capsule around the cell, which is to help prevent desiccation and to evade the immune system. And sometimes they'll have flagella. It might be one, it might be many, which is to help them to be able to move. But those aren't always found. Those are only sometimes found in prokaryotic organisms. Viruses are acellular and non-living. The structure of virus particles consists of genetic material, capsid, and attachment proteins, which we can see over here. So we have the genetic material in the middle. We have the capsid, and then we have the attachment proteins, or here they're labelled envelope proteins. Viruses replicate inside of host cells, and that is why it's very difficult to destroy them, because you would have to destroy the host cell to be able to destroy the virus. Now you need to know methods of studying cells, and the reason this is so important is the way we've discovered these internal structures or organelles is because of these advances in the methods to study cells. So advances in microscopes, and then also cell fractionation and ultracentrifugation to be able to isolate particular organelles to examine. So we'll go through each of these methods of how to study cells. First of all, with the microscopes, there's a couple of key definitions you need to be aware of. You also need to know the three main types of microscope. There are optical microscopes, which are the light ones, which you'd be using in school. You then have electron microscopes, but you can have either a transmission electron microscope or a scanning electron microscope. The definition of magnification is how many times larger the image is compared to the object. Resolution of a microscope is the minimum distance between two objects in which they can still be viewed as separate. And the resolution of an optical microscope is determined by the wavelength of light, and the resolution of the electron microscopes is determined by the wavelength of the beam of electrons. And because that wavelength is much shorter, that is why they have a higher or a more powerful, that is why they have a much, much higher resolution, meaning that the distance between the two objects is smaller. So let's compare the two general types of microscopes in more detail. The optical microscope, we've already said it's the light, but it is a beam of light. It's a beam of light that is condensed to create the image. In contrast, an electron microscope, it's a beam of electrons which is condensed to create the image. And what is used to condense the light and the electrons is also different. The beam of light is condensed using a lens, whereas the beam of electrons is condensed using electromagnets. The optical microscope has a poorer resolution due to the fact that light has a longer wavelength. So that means that the electron microscopes, because electrons have a shorter wavelength, they have a much higher resolving power. There's also a lower magnification for the optical, higher for the electron microscopes. You can have colour images though, and you can view living samples with the optical microscopes. So those are two big advantages. The electron microscopes can only produce black and white images, and because electrons can be easily absorbed, you have to have your sample in a vacuum. And because it's in a vacuum, you can't actually observe any living samples using an electron microscope. Now, just to go back to this black and white image point, you can still get coloured images if colours are added on programs like Photoshop afterwards. Because the optical microscopes have a lower resolution, small organelles in a cell are not visible. So you can't see details of the mitochondria, chloroplasts, ribosome. You can just see some of the large organelles, like we can see here. You can see the nucleus, you can see the DNA, you can see the chromosomes. The electron microscopes, they have much higher resolving powers, as we said, and the difference between the transmission and the electron is that the transmission, you have extremely thin specimens, and the electron gun produces a beam of electrons that transmits or passes through the specimen. So some parts of the specimen will absorb the electrons and therefore appear dark, some parts won't and the electrons would have passed through and they appear lighter. And we get these 2D images where we have the darker and lighter depending on whether the electrons were absorbed or not. And because there's a higher resolution, that is why we can actually see the details internally of some of those small organelles. A scanning electron microscope creates 3D images in contrast. The specimen does not need to be thin, and that's because the electrons are not going to be passing through the specimen. Instead, the electrons are beamed onto the surface, and the electrons are scattered in different ways depending on the contours. And that is how we get these 3D images, which we can see here in these different blood cells in a sample from the blood. You could be asked to calculate magnification of an image, so the formula that you need to remember is IM, and that is the image size equals actual size times magnification. Or you can rearrange it to work out the actual size or the magnification. And the key thing that you need to be aware of when you are doing these calculations is the image size and the actual size have to be in the same units. So you need to be familiar with how you convert from millimeters to micrometers or micrometers back into millimeters. Those are the two most common conversions in a magnification question. But if you want to know any of the other conversions, here they are. You might also need to be able to measure the size of a specimen using the eyepiece graticule. And inside of the optical microscope, there is a scale on a glass disc, which is called the eyepiece graticule. So it'd be just here within that eyepiece. And this can be used to measure the size of objects you're viewing under the microscope. However, each time you change the objective lens and therefore the magnification, you have to recalibrate the eyepiece to work out what the distance between each of these different divisions is actually worth. Now you need to know how to do that, but I'm not going to go through that in the summary video, but I'll link the video up here of how to calibrate the eyepiece graticule. Cell fractionation is the final way that you can study cells, and this is used to isolate the different organelles so that they can be studied further. First thing that happens is the cells are broken open to release the contents and the organelles. The cells need to be prepared in a cold, isotonic and buffered solution, and you need to know why it has to be those three conditions. So it has to be cold to reduce enzyme activity, and that's because when you break open the cell, you'll be releasing enzymes which wouldn't typically be in contact with the organelles and they might damage them. It has to be isotonic so that you don't have excess water moving in or out of the organelles by osmosis, because we don't want the organelles to burst or shrivel, because then we won't be able to study them. And it has to be buffered because if it became too acidic or alkaline, again, it could damage the organelles. Now, cell fractionation is a two-step process: homogenization and ultracentrifugation. So first of all, homogenization is when you break open the cells, and that can be done in a blender. As long as you have, as we said, the solution that you're blending up your sample in, as long as the solution that you're blending your sample in is cold, isotonic and buffered. Then we'd need to filter to remove the large debris, and then the filtrate, we can then use ultracentrifugation to isolate the different organelles. So we'd put our sample into a centrifuge and we'd spin at different speeds. The organelles are going to separate according to their densities, and that is why we have to spin our sample at different speeds. And this is what differential centrifugation is. So when we spin our sample, the centrifugal forces cause pellets to form at the bottom with the most dense organelles. So we can see here in this first image, we've got all of the organelles equally distributed. And if we first spin at low speed, the most dense organelle will form a pellet at the bottom. We would then remove that pellet and spin the rest of the filtrate again at a slightly faster speed. We'd then get the next most dense organelle forming the pellet, and we repeat this process at increasingly faster speeds. So each time that liquid, which is the supernatant, is removed and the pellet is what the organelles will be in, and we can examine those. Now, just to let you know the order of the density, in that first centrifugation, the nuclei are the most dense. So those would be isolated in the first spin. The next most dense are the chloroplasts and the mitochondria, then the lysosomes, endoplasmic reticulum, and finally the ribosomes. So that would be the order in which you would collect your different organelles. We then move on to cell division, and eukaryotic cells enter the cell cycle and divide by mitosis or meiosis. But in topic 1, you only learn about mitosis. In comparison, prokaryotic cells replicate by binary fission, and viruses do not undergo cell division at all because they are non-living. They do still replicate though, but viruses replicate inside of a host cell. They will invade that host cell by injecting in their nucleic acid, so their genetic material, and then it will be the host cell that uses that genetic material to replicate the virus particle. The cell cycle that eukaryotic cells will be going through includes these key stages. Interphase is the longest stage of the cell cycle, and it includes G1, S, G2. G1 is when the cell is going to be increasing in size and the organelles will double. S phase is when DNA replication happens. G2, you'll have further growth, but also it says preparation for mitosis. In G2, you'll have this error check stage. So if there are any errors in the DNA replication, the cell would be destroyed at that stage. Nuclear division is either mitosis or meiosis, but in topic 2, we just focus on mitosis. The final stage of the cell cycle is cytokinesis, And this is when the cytoplasm divides to create the two new cells if it's mitosis. Mitosis is split into 4 key stages, which are our PMAT, prophase, metaphase, anaphase, and telophase. The key facts about mitosis are it's only one round of division, genetically identical cells are created, the cells are diploid, which means there are 2 copies of every chromosome, and this stage is used for growth and repair. So a specific example of that, which we'll see later in this video, is the clonal expansion of B cells. So that is growth in the sense of we're creating lots of new cells. So in prophase, the chromosomes will condense and at this point they become visible. And in the animal cells, the pairs of centrioles will move to opposite poles, which means opposite sides of the cells. The centrioles are going to be creating these spindle fibres, which are released from both poles to create the spindle apparatus. And these will attach to the centromere and the chromatids on the chromosomes in the later stages. Plants have a spindle apparatus, but they don't have the centrioles. In metaphase, we can see that the chromosomes will then line up in single file along the equator, and the spindle fibres are released from those centrioles at the poles, and they'll attach to the centromere and also the chromatids. In anaphase, the spindle fibres will start to retract and pull back towards the centrioles, and in doing that they'll pull on the centromere and chromatids, and this causes the centromere to divide in 2, and the individual chromatids are pulled to the opposite poles of the cell, and therefore it separates the chromatids, and once those chromatids are separated, we now actually call them chromosomes. Now this stage requires energy in the form of ATP, and that's provided by respiration from the mitochondria. In telophase, the chromosomes are now at each pole of the cell and become longer and thinner. The spindle fibres will then disintegrate and the nucleus starts to reform. The final stage in the cell cycle is when the cytoplasm splits in 2 to create the 2 new genetically identical cells, which the mitotic index can be calculated by counting first of all how many cells are visible in the field of view, and then counting the number of cells that are also visible but in a stage of mitosis. You can then do a percentage calculation. So you'd be doing the number of cells that you can see in a stage of mitosis divided by the total number of cells present and times it by 100. Prokaryotic cells don't go through mitosis, instead they go through binary fission. The first step of that would be the replication of the circular DNA and of the plasmids, and then the second stage would be division of the cytoplasm to produce 2 daughter cells, each with a single copy of the circular DNA and a variable number of plasmids. Viruses are non-living, so they don't undergo cell division. Instead, they inject their nucleic acid into the host, and the host cell replicates the virus particles. So now on to plasma membranes. All cells and organelle membranes have the same structure. It's this fluid mosaic model, which is named because of the fact that it does have some slight movement, and also it's composed of a range of different molecules, such as phospholipids, proteins, glycoproteins, and glycolipids. All of these molecules are arranged within the phospholipid bilayer, and they create the partially permeable membrane. To create the bilayer, the phospholipids align in this manner because of the fact that the phospholipid has a hydrophilic head due to that negative charge on the phosphate group, and that causes it to be attracted to water. Whereas the hydrophobic tails, those are repelled by water but can interact with lipids. So the phospholipids end up spinning round so that the tails are opposite each other, the heads are on the outside where they can interact with water. Cholesterol is present in some membranes too, and this will restrict the lateral movement of other molecules in the membrane. This is useful as it makes the membrane less fluid at high temperatures and prevents water and dissolved ions from leaking out of the cell. The other components of the membrane are mainly the proteins, and these are embedded across the cell surface membrane, either as peripheral, meaning they're just on the outside, sometimes called extrinsic, or they could be integral, sometimes called intrinsic, meaning it spans from one side of the bilayer to the other. The peripheral proteins provide mechanical support, or they can be connected to proteins or lipids to make glycoproteins and glycolipids. The peripheral proteins provide mechanical support, or they can connect to carbohydrates to make glycoproteins, and also carbohydrates can bind to the lipids directly to make glycolipids. The function of these is cell recognition or as receptors. The integral proteins are protein carriers or protein channels involved in the transport of molecules across the membrane. Protein channels are tubes, and these fill with water, which will enable water-soluble ions to diffuse across the membrane, whereas the carrier proteins will enable molecules to bind with them, and therefore larger molecules like glucose and amino acids can cause those carrier proteins to change shape and therefore transport the molecule to the other side of the membrane. So when we say that the membrane is partially permeable, what we mean by that is only lipid-soluble substances and very small molecules can pass across the membrane by simple diffusion. Other molecules, such as water-soluble or polar substances and large molecules, can't simply diffuse across the membrane. Therefore, they have to be transported across by other means, which could be facilitated diffusion, active transport, or if it's water, osmosis. So simple diffusion, first of all, this is the net movement of molecules from an area of higher concentration to an area of lower concentration, and this will continue until an equilibrium is reached, and this process does not require any ATP. The molecules do still have energy to be able to move, but this is just due to the kinetic energy that they possess, and that is why they're able to move constantly as a fluid. For molecules to diffuse across the membrane, they have to be lipid soluble and small. If they're not lipid soluble or small, then it will be facilitated diffusion instead. And this is still a passive process because the molecules are moving from an area of higher concentration to lower, but because they can't simply diffuse through the phospholipids, instead they have to diffuse through proteins. Now this could be through protein channels, where we have those tubes filled with water, and therefore water-soluble ions can dissolve and then diffuse through. This is still selective though, as the channel proteins only open in the presence of certain ions when they bind to the protein. Carrier proteins will bind with a molecule such as glucose, which causes a change in the shape of the protein. This change enables the molecule to be released to the other side of the membrane. Osmosis is the movement of water, and that will be from an area of higher water potentials to an area of a lower water potential across a partially permeable membrane. The water potential is the pressure created by water molecules, and it's measured in kilopascals and represented by this symbol here. Pure water or distilled water has a water potential of 0. So you cannot get one any higher than 0. It's always going to be 0 or negative. And that means when solutes are dissolved in water, the water potential will be negative. And the more negative the water potential, the more solutes there must be dissolved in it. Now, 3 key terms that you need to know linked to osmosis are isotonic, hypertonic, and hypertonic. An isotonic solution is when the water potential is the same in the solution and the cell. Hypotonic is when the water potential of the solution is more positive, so closer to 0 or closer to pure water compared to the cell. Hypertonic is when the water potential of the solution is more negative than the cell, so there'd be more solutes dissolved in it. And in an animal cell, we can see the impact that would have. In an isotonic solution, because the water potential is the same, there's not going to be any net gain or loss of water. In a hypertonic solution, there'll be more water moving out of the cells by osmosis, and that can cause animal cells to shrivel or crenate. If animal cells, such as red blood cells, are put into a hypotonic solution, That would mean water would be moving into the cell by osmosis, and if enough water moves in, it can cause the cell to burst. Active transport is the movement of substances from a lower concentration to a higher concentration, and this requires metabolic energy and a carrier protein. So the process by which this happens is, first of all, it's always through carrier proteins only. It is not channel proteins for active transport. The molecule will bind to the carrier protein, and it can only bind to proteins which have a complementary receptor shape, so it is still selective. The ATP binds to the carrier protein as well from the inside of the cell, and it is hydrolyzed into ADP and PI. This causes the carrier protein to change shape, and therefore it releases the molecule to the other side of the membrane. The phosphate ion is then released, and the protein can return to its original shape, and the process can then happen again as long as there is plenty of ATP present. Co-transport is a type of active transport, and you learn about it across the course, but one of the key examples you learn about is the co-transport of glucose, or it could be amino acid, with sodium ions in the ileum. And to absorb glucose from the lumen of the gut, there has to be a higher concentration of glucose in the lumen compared to the epithelial cell. But there's usually more glucose in the epithelial cells, and that is why active transport and co-transport are required. So the first thing that has to happen is the sodium ions, which are shown in blue here, are actively transported out of the epithelial cell into the capillaries. And that will result in a lower concentration of sodium ions within the epithelial cells. And that creates this concentration gradient from the ileum to the epithelial cell. And therefore the sodium ions can diffuse down their concentration gradient into the epithelial cells. The proteins the sodium ions diffuse through, though, is a co-transporter protein. So either glucose or amino acids will also attach and are transported into the epithelial cell with the sodium ions, except the glucose is going in against its concentration gradient. The glucose, once in the epithelial cell, can then move by facilitated diffusion from the epithelial cell into the blood. Now, you also need to be aware that cells may be adapted for rapid transport across their internal or external membranes. So it could be that cells have an increased surface area, so things like microvilli, for example. Or there could be an increase in the number of protein channels and carrier molecules within the membrane as another way to increase the rate of transport. The last section of topic 2 is all about immunity, and we begin by looking at identifying self and non-self cells. So your body's immune system has cells to be able to identify the presence of pathogens and potential harmful foreign substances compared to your own body cells. And that's so it can destroy the foreign items and not your own cells. And those cells are called lymphocytes. But how can lymphocytes then actually distinguish between a pathogen and your own body cells? Well, each type of cell has specific molecules on its surface that identify it. And these molecules are usually proteins, so they'll have this unique 3D tertiary structure. And this is how they can identify whether it is an antigen from a pathogen or whether it is one of your self-antigens. If a non-self cell is detected, a response will be triggered to destroy that cell. And these different surface molecules enable them to identify pathogens, which we've mentioned, so bacteria, fungi, viruses, but it could also be cells from other organisms of the same species. And that can be harmful for individuals who have had organ transplants. It could also be abnormal body cells, for example, cancer cells. And finally, toxins, because some pathogens release toxins into the blood, such as the bacteria cholera. Now, an antigen is a molecule that generates an immune response. and this is because it triggers the lymphocytes. They are usually proteins and they are located on the surface of the cells. Now, antigen variability is this concept that pathogens' DNA can mutate frequently. If a mutation occurs in the gene which codes for the antigen, then that means the shape of the antigen on the outside of the virus, the fungus, the bacteria will change. And if that happens, any previous immunity that you may have had to the pathogen is going to be lost. So it will no longer be effective, as all of the memory cells in the blood that you have will have a memory of the old antigen shape and not the new one. And this is known as antigen variability. The influenza virus mutates and changes its antigens very quickly, and this is why a new flu vaccine is created every year to account for that change. So looking at your immune response then, if a pathogen gets past the chemical and physical barriers, so for example your skin is a physical barrier and stomach acid is a chemical barrier, then your white blood cells are the next line of defense. Your white blood cells have a specific and also non-specific response. The phagocytes are a non-specific response, meaning that they will destroy any foreign item that they come across. Lymphocytes have a specific response though, and this is where the idea of detecting using antigens will come into it. So let's look at phagocytosis first of all. A phagocyte is a macrophage cell, which is a type of white blood cell, and they are found in the blood but also tissues. Phagocytosis, as we said, is a non-specific response. So any non-self cell that is detected will trigger the same response every time to cause the destruction of that object. So the stages of phagocytosis are, first of all, the phagocytes are in the blood and the tissues, and any chemicals or debris released by the pathogens or abnormal cells attract the phagocytes, and that will cause those white blood cells to move towards the cell. There are many receptor binding points on the surface of the phagocyte, which we can see here. They will attach to the chemicals or the antigen on the pathogen via the receptors. The phagocyte then changes shape to move around the pathogen and engulf it, and once engulfed, the pathogen is contained within a vesicle, which we call a phagosome. A lysosome within the phagocyte will then fuse with the phagosome and it will release its contents. And the contents is a lytic enzyme called lysozyme. And lytic enzymes are able to hydrolyze. So the result is that that enzyme will hydrolyze and it breaks down the pathogen. Any soluble products are absorbed and used by the phagocyte, and anything else is released as waste and debris. So that is our non-specific response, but now if we look at the specific responses, which are by the lymphocytes, first lymphocyte we're going to look at are the T lymphocytes, or the T cells. So all lymphocytes are made in the bone marrow, but the T cells mature in the thymus, which is why they're called T cells. And this is classed as the cell-mediated response. So the first thing to be aware of is the cell-mediated response is specific to T cells responding to antigens on the surface of cells. So it involves antigen-presenting cells, or APC. An antigen-presenting cell is any cell that presents a non-self antigen on their surface. Infected body cells will present the viral antigens on their surface if they are infected. A macrophage, which has engulfed and destroyed a pathogen, will also present the antigens on their surface. Cells of a transplanted organ will have different shaped antigens on their surface compared to your own self cells. And cancer cells will have abnormal shaped self antigens as well. So all of these are presenting antigens on the cells, so they are antigen presenting cells and can therefore trigger an immune response. So the cell mediated response then, once a pathogen has been engulfed and destroyed by a phagocyte, the antigens are positioned on the cell surface. and that is why we'd now call that phagocyte an antigen-presenting cell. Helper T cells, which are a type of T lymphocytes, have receptors on their surface that can attach and bind to the antigen on antigen-presenting cells. And once those two cells are bound, it activates the helper T cell to start to divide by mitosis. And that then means we get a very large number of cloned cells. So essentially, this stage is to make sure you end up with lots of helper T cells which have the correct shape to bind on to that particular antigen. So you'll have lots of helper T cells within your blood that have different shaped receptors to be able to bind onto different shaped antigens. But if you have one collide, that will then activate that particular T cell with that particular shape receptor to divide by mitosis to make large cloned copies. Those cloned helper T cells then differentiate into particular types of T cells. Some remain as helper T cells, which then go on to activate the B lymphocytes. Some can stimulate macrophages to perform more phagocytosis. Some become memory cells for that particular shaped antigen. And some become cytotoxic killer cells or cytotoxic T cells. And what those cytotoxic T cells do is destroy the abnormal or infected cells that have the antigen on their surface. And they do this by releasing a protein called perforin, which embeds in the cell surface membrane to make a hole or a pore, so that any substance can now enter or leave the cell. Now, this causes death because you can have lots of water move in and therefore the cell will burst, or you could have lots of water moving out and therefore the cell will actually shrivel up and die. This is most common in viral infections because viruses infect body cells of the host. Body cells therefore have to be sacrificed to prevent further viral replication. And this is actually why you get a really sore throat when you have a cold, because those cytotoxic T cells are destroying the infected body cells in your throat to try and prevent that virus dividing and spreading further in your body. Next then, we move on to the B lymphocytes or the B cells. As we already said, all lymphocytes are made in the bone marrow, but B cells mature in the bone marrow as well, which is why they're called B cells. This is now called the humoral response, and this is the one that involves antibodies. Antibodies are soluble and transport in bodily fluids, and humour is an old term for body fluids, and that's why this is called the humoral response. There are approximately 10 million different B cells which have antibodies on their surface, complementary to 10 million different antigens. And antigens in the blood will collide with their complementary antibody on a B cell, and the B cell takes in the antigen by endocytosis and then presents it on its cell surface membrane. When that B cell collides with a helper T cell, that can then activate the B cell to go through clonal expansion and differentiation or clonal selection. So this is the step where there's that link between the cell-mediated response with the helper T cells binding via the receptor to the antigens on the B cells to activate them. Once activated, the B cells undergo mitosis to make large numbers of the cells which have that particular antigen on their outside. And these can then differentiate into plasma cells or memory B cells. The plasma cells will go on to make antibodies complementary in shape to that particular antigen. And the memory cells can divide rapidly into plasma cells if you are reinfected with the same pathogen and therefore antigen later on. And that means that you can make large numbers of the correct shaped antibody so rapidly that you should be able to destroy the pathogen before it causes any damage and symptoms. So the memory B cells can live for decades in your body, whereas the plasma cells are only short-lived. Memory B cells, though, cannot make antibodies, but they can divide by mitosis and then differentiate into plasma cells. So this is what results in the large numbers of antibodies being produced so rapidly that the pathogen is destroyed before any symptoms occur. And that's what it means when we say you are immune to a particular disease. It's because you have these memory B cells, so you can destroy the pathogen before it causes any symptoms. And that would be an example of active immunity. Primary and secondary response is referring to the number and the speed at which antibodies are produced when you are first exposed to an antigen compared to your second exposure. So the first time you're exposed to a new pathogen and therefore a new antigen, it takes a little bit longer for the antibodies to be produced because you have to go through that initial step of those 10 million different B cells colliding until you have the correct shape antigen-antibody binding. So that can take a couple of days, and that's why you have a slower response in producing the antibodies, and also you don't actually make as many. But because memory B cells are made, if you are exposed for a second time, you will be able to create those antibodies very, very rapidly and in much larger numbers. And this is what is meant by the primary response and the secondary response. So if we look a bit more at antibodies, antibodies are an example of quaternary structured proteins because they're made up of 4 polypeptide chains. The part shown in purple is described as the variable region, and that is where you would have the part of your antibody that binds onto particular shaped antigens. The part in red is constant, so it would be the same for different antibodies. We can then see we have one longer chain, which is heavier, so we call it the heavy chain, and we have a shorter chain, which is called the light chain. And this bit here where it is variable, as we said, that is the antigen binding site. Now those antibodies are actually flexible, and that enables an antibody to bind to multiple antigens. So we can see we've got binding to 2 antigens here, and then this antibody is binding to 2 as well, and as a result they all end up clumping together, and we call this agglutination. The advantage of agglutination is if you've got a big clump of antibodies and antigens which are going to be attached to the pathogen, it makes it much easier for the phagocytes to locate and therefore destroy the pathogens more rapidly. So we've talked about active immunity. Passive immunity, though, is when the antibodies are introduced to the body. So you haven't made the antibodies yourself, you are just gaining the antibodies. So that means that the pathogen didn't cause the creation and differentiation of plasma cells or any memory cells, so you won't have any long-term immunity. Examples of passive immunity could be antibodies passed to a fetus through the placenta or through breast milk from the mother to their baby. Active immunity, as we said, is when the immunity is created by your own immune system following the exposure to the pathogen. But this can be either natural or artificial. And natural is following an infection where you've actually had that pathogen within your body. Artificial is following the introduction of a weakened version of the pathogen or antigens via a vaccine. So if we go through then how vaccines work. Small amounts of weakened or dead pathogen, or it could just be the antigens, are introduced either to the mouth or injected into the body. And exposure to those antigens activates the B cells to go through clonal expansion and differentiation. So that was the clonal selection part where we have mitosis occurring to make large numbers of those B cells. Those then differentiate into plasma cells and memory B cells. As we said earlier, the plasma cells will make antibodies, but the key bit of importance for a vaccine is the fact that B memory cells will also be produced, and those can stay in the blood for years. So that means that if you are infected with the actual pathogen, those memory B cells, if they collide with the antigen, will divide rapidly into plasma cells when you are reinfected, and therefore you create large numbers of antibodies so rapidly that you shouldn't get the symptoms of the disease. Or if you do, they'll be very, very mild and you should be able to overcome the disease more quickly. Now, not everyone is able to take vaccines for various reasons, but the concept of herd immunity is if enough of the population are vaccinated, the pathogen cannot spread easily amongst the population anymore. So this provides protection for those who aren't vaccinated. For example, if you already have another illness, which means it'd be too dangerous for you to have the vaccine, if you have a lowered immunity, if you're too young, and sometimes if you're pregnant, you're not able to take vaccines as well. Now, the structure of HIV is made up of 4 key components. We have the core, and that is the genetic material, which is RNA, and the enzyme reverse transcriptase. And this is needed for the viral replication when it's in the host. You also have a capsid, which is an outer protein, which you can see just here. You then have the envelope, which is an extra outer layer made out of the membrane taken from the host cell membrane. And then we have protein attachments on the exterior of the envelope to enable the virus to attach to the host's helper T cells. So the way that HIV replicates in helper T cells is that first of all, it's transported in the blood until it attaches onto a CD4 protein, which is one of the attachment proteins on the outside of helper T cells, which we can see just here. The HIV protein capsule then fuses with the helper T cell membrane, enabling that RNA and enzyme to be injected or enter into the host's cell. The HIV enzyme reverse transcriptase copies the viral RNA that's been injected into a DNA copy and moves to the helper T cell nucleus, and this is why it's called a retrovirus. Here, the mRNA is transcribed and the helper T cell starts to create viral proteins to make the new particles. Someone's described as HIV positive when they are infected with HIV. AIDS is when the replicating viruses in the helper T cells interfere with the normal functioning of the immune system. So someone who is HIV positive doesn't necessarily have AIDS. With the helper T cells being destroyed by the virus, the host is unable to produce an adequate immune response to other pathogens and is left vulnerable to infections and cancer. It is the destruction of the immune system that leads to death rather than the HIV directly. So the very final thing is this concept of monoclonal antibodies. Mono meaning one and clonal meaning identical. Antibodies are proteins, which we looked at, and they have these binding sites complementary in shape to a particular antigen. And this has been manipulated to create monoclonal antibodies. And that can be for medical treatments, medical diagnosis, it's used in pregnancy tests, it's also used in tests such as drug testing and testing for other viral diseases. So targeted medication, we're going to go through that example first. This is an example of direct monoclonal antibody therapy. So what we mean by that is some cancer can be treated using monoclonal antibodies, which are designed with a binding site complementary in shape to the antigens on the outside of the cancer cells. The antibodies are given to the cancer patient and they will then attach onto the cancer cells only. While the antibodies are bound to the cancer cells, this prevents chemicals binding to the cancer cell, which will enable uncontrolled cell division. Therefore, the binding of these monoclonal antibodies prevent the cancer cells from growing. And as they're designed to only attach to the cancer cells, they're not going to harm any other normal cells. Indirect monoclonal antibody therapy can also be used to treat some cancers. The monoclonal antibody is created exactly the same, except there will be a drug attached to them. Indirect monoclonal antibody therapy starts with the same process of creating a monoclonal antibody which has a binding site complementary in shape to the antigens on the outside of cancer cells. but this time a drug is also attached to the antibody. The cancer drugs are therefore delivered directly to the cancer cells and can destroy those cells only. And this reduces the harmful side effects that traditional chemotherapy and radiotherapy can produce. And this is sometimes called a bullet drug because it's being directed to just one particular site. Medical diagnosis is another use, and as we said, this could be in pregnancy tests, viral tests, so influenza, could be in bacterial tests as well, cancer tests. Most recently, we've seen in COVID-19 lateral flow tests as well. And these use the ELISA or the ELISA test. So the ELISA test, which stands for Enzyme-Linked Immunosorbent Assay, or ELISA, uses two antibodies. So first of all, the first mobile antibody, which is complementary to the antigen being tested for and has a coloured dye attached to it, we can see just here. Part C, we have a second antibody, which is complementary in shape to the antigen as well, but this one is immobile, so it's attached to this fixed position. There is then a third antibody, which is immobilized at point D, and it is complementary in shape to the first antibody. So what this means is, if you are pregnant, if we think about this in terms of a pregnancy test, you contain the hormone HCG within your urine. That will then bind to the antibody, and any antibodies with the HCG hormone bound to it will then attach to the immobilized antigen at point C. And therefore, you get a coloured blue line at this point because we have that blue bead held in that fixed position. You should always have a second blue line, whether you're pregnant or not, at position D though, because this second immobilized antibody is just complementary to the shape of the first one. So it's basically just a control to check that you do definitely have that first antibody in your sample and it is able to move along the test kit. Now, another test we can look at is this one here for the ELISA test, where you would add the test sample from a patient to the base of a beaker, wash to remove any unbound test sample. Then we add an antibody complementary in shape to the antigen you are testing the presence of in the test sample. Again, we then wash to remove any unbound antibody. Add a second antibody that is complementary in shape to the first antibody and binds to the first. The second antibody we can see here has an enzyme attached to it. Again, we have to rinse just to make sure if there's any unbound antibody, those are taken off. And then the substrate for the enzyme, which is colourless, is added. And this substrate produces a coloured product when that enzyme is present. So the presence of colour indicates the presence of the antigen in the test sample, and the intensity of the colour indicates the quantity present. Now, there are some ethical issues surrounding monoclonal antibodies, and that is because within the creation of monoclonal antibodies, you do require animals such as mice to produce the antibodies and tumour cells. And that can lead to ethical debates as to whether the use of animals is justified to enable the better treatment of cancer in humans and to detect diseases, because this will cause discomfort, harm and death to the animals that are being used. So we begin with surface area to volume ratio, and this is where we have this particular formula of whatever the surface area of an organism is, you divide it by its volume. And the relationship between how large an organism is, or it could be the different shapes it has, are linked to the surface area to volume ratio. So this concept has a big role within this topic, where we look at how different cells or even organ systems have adaptations to maximise gas exchange and other types of transports across surfaces. So here we just have an example where we can see this pattern of the larger an organism is. Now in this case we've just got cubes to demonstrate, but the larger an organism is, the surface area divided by the volume, or the surface area to volume ratio, will decrease. So what that means in terms of biology is small organisms like single-celled organisms like amoeba have very large surface areas compared to their volume. And that means that they don't have to have any additional special adaptations to meet their needs in terms of transport across their surface. They can just diffuse gases across their surface, and because they've got such a large surface area compared to their volume, they'll be able to get enough gases in and out to meet their metabolic needs. However, larger organisms, because the surface area is so much smaller compared to the volume, that means that if we were just to get gases into the organism through simple diffusion, the cells right in the centre of the organism are so far away that the gases wouldn't be able to reach there in sufficient time for their metabolic needs. Now, on top of that, larger organisms typically have higher metabolic needs as well. And what that means is the chemical reactions within a cell are happening faster. So that is another reason why large organisms have to have adaptations for mass transport or just for transport across cells. And that's what most of this topic is looking at, the adaptations that organisms have for their exchange surfaces to make exchange more efficient. So some of the key adaptations are the villi and the microvilli. So within the small intestines, those two particular types of tissue and then cells to maximize the absorption of digested food. The alveoli and bronchioles, so they're involved in gas exchange. Spiracles and tracheoles involved in gas exchange, but that's for terrestrial insects, whereas alveoli and bronchioles are for mammals. Gill filaments and lamellae is the gas exchange adaptation surface for fish. And then we'll be looking at plants as well. They exchange gases through the stomata on the leaves, but we'll be looking at some of those adaptations as well. Now, in addition to that, when we get to the idea of the mass transport as well, we'll see a link to gas exchange, whereby most of these gas exchange surfaces have a capillary network nearby as well, with the exception of plants because they don't have blood. So some of the key terms linked to the gas exchange and ventilation topic that you need to be familiar with are breathing. So this is the movement of air in and out of the lungs. This is different to respiration because respiration is a chemical reaction that results in the release of energy in the form of ATP. Ventilation is the scientific term for breathing, so we'll be using the term ventilate quite a lot in this video. And gas exchange is the diffusion of typically oxygen and carbon dioxide in and out of cells. Now, it's not just the alveoli, that is just for mammals. So those are the key terms just to be familiar with, and these are the key structures that you need to know for the human gas exchange system. So the alveoli, bronchioles, bronchi, trachea and lungs, which are all labelled here. So if you need to pause just to note that down, then pause that just here. So we have a look at how humans ventilate. It involves the muscle, which is the diaphragm. It also involves antagonistic muscles, which are surrounding the ribs. And this is where we have the external intercostal muscles, but we also have internal intercostal muscles. Now, antagonistic means that as one muscle is contracting, the other relaxes. And we'll see that in our intercostal muscles, how the internal and external will always be doing the opposite. And that is what controls the movement of the rib cage in and out to help with ventilation. So a bit more about these internal and external intercostal muscles. Antagonistic muscles always occur as pairs, and the external intercostal muscle on the outside layer of the ribs, and they will contract and cause the rib cage to move out and air to flow in, or inspiration. The internal intercostal muscles, you can kind of see as it moves around 3D, they'll be on the inside layer around the ribs. And when these ones contract, that will pull the rib cage closer inwards, and therefore the air will flow out, or you'll be expiring. But just to have a summary then of what is happening to all of the muscles that are controlling the movement of the rib cage, and therefore whether you're inhaling or exhaling or inspiring or expiring. So when you are breathing in, that is when your external intercostal muscles will be contracting, and that pulls the rib cage up and outwards. That means that the internal intercostal muscles are doing the opposite, they're relaxing. The diaphragm muscle contracts, and when that contracts, it causes it to move downwards and it becomes more flat. Now, as a result, that means that the volume of the thorax will be larger, so the volume increases. And by that, we don't mean the volume of gases, we mean the volume as in the cavity space, so the available volume, the actual space available. And because the lung volume, the thorax space, is larger, that causes the pressure to drop. And because you now have a lower pressure within the lungs compared to outside, air is going to flow in. So that is what happens when you breathe in. Now, when you exhale, it's exactly the opposite. So this time the external intercostal muscles relax, the internal will contract, and that pulls the rib cage down and back in. The diaphragm relaxes, which causes it to dome upwards. And as a result, the actual volume in the thorax, that space, is smaller. And because we have a smaller space or a smaller volume, that causes the pressure to increase. And that is why the air is forced out of the lungs. Now, the pulmonary ventilation calculation is one that you do need to know, and it's the total volume of air that's moved into the lungs during 1 minute. And the units would be decimeters cubed for volume and per minute. So to work this out, you would be doing the tidal volume times the ventilation rate. So tidal volume is the volume, total volume of air, and the ventilation rate is how many times you breathe per minute. So then if we go on to the gas exchange and look at the alveolar epithelium. Once the gases are in the alveoli, so as a result of ventilation, this is where gas exchange occurs, and it's between the epithelium and the blood. Alveoli are tiny air sacs, and you have around 300 million in each lung. And that's actually why lung tissue floats compared to, let's say, muscular heart tissue, which would sink. So all of those alveoli air sacs make it float because it's so full of air. Now, the alveoli epithelium cells are also very, very thin. And that is to minimize the diffusion distance. So we can see here there's just one layer of cells and they're a thin layer. Each alveolus is surrounded by a network of capillaries, and this helps to maintain the concentration gradient, because as soon as that oxygen diffuses into the blood, it is then carried away in the capillaries and replaced by deoxygenated blood. Terrestrial insects, they do have a large surface area to volume because they're small. However, the issue they have to overcome is the fact that they have a waterproof exoskeleton. And that's an advantage to prevent water loss, but because it's made up of this hard fibrous material, it also means gases can't exchange across it. Now, insects don't have lungs, but they do have a tracheal system, which is where ventilation and gas exchange occurs. So their tracheal system then, we have a look at how this differs to the human lungs. First of all, it includes a trachea, tracheoles and spiracles. The spiracles we can see here, they're the tiny holes, they're round valve-like openings that run along the abdomen, and this is where oxygen and carbon dioxide are going to be entering and leaving. So that is instead of like the nose or the mouth for humans. And those spiracles are going to be attached to trachea. So that's the next structure. And the trachea are tubes. They have rings within them to strengthen the tubes to stop them from collapsing, and that keeps the tubes permanently open. And then the trachea branch into smaller tubes, which are deeper in the abdomen of the insect, and those are called the tracheoles. And those extend throughout all of the tissue in the insect to deliver oxygen to all of the respiring cells. So here we have the spiracles connected to the trachea, and then the tracheal systems branching all the way through into the centre where the abdomen is, and to all of the respiring tissues. Now there's three methods of moving gases into that tracheal system. The first method is gases can just move in by diffusion. And that is because when the cells within the insect are respiring, they're using up oxygen, producing carbon dioxide, and that creates a concentration gradient from the tracheoles compared to the atmosphere. So some of the gases will simply diffuse in and out. The second method of gas exchange is mass transport. And this is where it's slightly similar to ventilation in humans, in that the abdominal muscles can contract and relax. And that is a way to ventilate, to move gases en masse in and out of the tracheal system. The final method is when the insects are in flight, the muscle cells will actually start to respire anaerobically. And when they do that, they produce lactate or lactic acid. And this lowers the water potential of the cells, and as a result, it draws water into the cells from the tracheoles. That decreases the volume in the tracheoles, and as a result, more air from the atmosphere is drawn in. So overall, the adaptations that terrestrial insects have for gas exchange are the fact that they have a large number of these fine tracheoles, providing a large surface area. The walls of the tracheoles are very thin, and there's a short diffusion distance between the spiracles and the tracheoles, so that will speed up the rate of diffusion. And lastly, they use oxygen and they produce carbon dioxide, and that is how the steep concentration gradient is maintained. Now, the next challenge that the terrestrial insects have to overcome is limiting the amount of water that is lost at these gas exchange surfaces, because a gas exchange surface is also ideal for evaporation, and they do not want to be losing excess water. So here are the ways that water loss is prevented. Number one is the surface area where gas exchange actually happens is very small. So it's not the entire insect, we're just looking at the spiracle itself and then the tracheal system. That is a very small area compared to the whole volume of the insect, and therefore it's going to limit evaporation. The rest of the insect has this waterproof exoskeleton, and the spiracles can actually open and close, a bit like stomata on a plant, to reduce water loss also. So next is gas exchange in fish. Now fish are waterproof and they have a small surface area to volume ratio. So those are the two reasons why fish have to have a gas exchange surface with adaptations. And for fish, it's gills. Now fish obtain their oxygen from the water, but there is 30 times less oxygen dissolved in water than there is available in the atmosphere, in the air. So they have to have an additional adaptation to help maintain the diffusion and concentration gradient across their gills. So just as a reminder, for all of these gas exchange surfaces, they have to have these three features: a large surface area compared to the volume, a short diffusion distance, and they have to have a mechanism to maintain the concentration gradient. Now those features can be used to work out the rate of diffusion using Fick's law. And this is just here. So diffusion is proportional to the surface area times the difference in concentration divided by the length of diffusion pathway. So you could get questions linked to this, testing that math skill. But let's have a look then at the fish gills to see how they have these three features maximised in their adaptations. So we're going to have a look at the fish gill anatomy. So there are 4 layers of gills on both sides of the head, and the gills are made up of stacks of gill filament. Each gill filament is covered in gill lamellae, and these are positioned at right angles to the filament. So this here is the lamellae, and this part is the filament. Now because there are so many, that creates the large surface area. So that's the first adaptation ticked off. Because there's so many gill lamellae and gill filaments, it creates that large surface area. Now when a fish opens its mouth while it is swimming, water rushes in and then actually comes out the side of their head, which means that the water rushes over the gills. So the water enters here and then it goes over the gills on both sides of the head, where they have these 4 layers of gills with the gill filaments and the gill lamellae. So the next thing is looking at the additional adaptations. We've talked about the large surface area to volume ratio, but the next thing is the fact that there is a short diffusion distance, and that is because of the network of capillaries in every gill lamellae. So we're zooming in here on the gill lamellae, and there is our network of capillaries within each one, and that provides a short diffusion distance. How the concentration gradient is maintained is by something called the counter-current flow mechanism. So the counter-current flow mechanism is where water flows over the gills in the opposite direction to which the blood is flowing through the capillaries. The reason this is an advantage is it makes sure that the equilibrium of the concentration of oxygen is never reached. And because we don't have equilibrium being reached, that means that oxygen is able to continue diffusing from the water into the capillaries in the gill lamellae across the entire gill lamellae. And that's in bold and underlined because that is the key marking point. Now that's demonstrated in these two diagrams here where we look at concurrent flow in comparison to countercurrent flow. So in concurrent, not co-current, you'd have the water and the blood flowing in the same direction. So about halfway across the lamellae, you'd be reaching equilibrium, and therefore you wouldn't have any more diffusion of gases. Whereas here we have the water and the blood flowing in opposite directions, which means you should never actually have equilibrium, and there will always be a higher concentration of oxygen in the water compared to the blood, and that is why we maintain the concentration or the diffusion gradient across the entire gill lamellae. So lastly is looking at gas exchange in leaves. So you could be asked to look at some of the structures in the leaf. So we have the tissue layer palisade mesophyll, which is where photosynthesis mainly happens. And then we have the spongy mesophyll, where there's lots of air spaces. And we have the stomata, which is where the gases actually diffuse in and out of. So oxygen will actually diffuse out of the stomata if it's not being used in respiration. And carbon dioxide diffuses in because it's needed for photosynthesis. To reduce water loss by evaporation, stomata close at night when it's dark, and they'll open in the daytime when it's bright. And this is linked to the idea of photosynthesis because it's a light-dependent reaction. Now there are compromises though, and in particular linked to xerophytic plants, which are plants that are adapted to survive in environments with very limited water. So they have adaptations to minimise water loss because there's such little water available already. And over here, looking at this marram grass under the microscope, it demonstrates some of those adaptations really well. So the leaves aren't actually flat, they roll up, and we have stomata which are really deep and sunken in, and we have lots of tiny hairs sticking out. Now, all of those adaptations are to reduce water loss because any water that is evaporating out is going to get trapped on these hairs, and the fact it's curled in, it gets trapped in this space. So it makes this area very, very humid, and if it's very humid, that will reduce further evaporation. Now, in addition to that, they also have a thicker cuticle to reduce water loss, and we can't see it on this particular image, but they do have a longer root network to be able to reach further distances to try and find any available water. So we now move on to digestion and absorption. And during digestion, large biological molecules are hydrolyzed into smaller soluble molecules which can be absorbed across the cell membranes. Now there's 3 biological molecules that you need to know the digestion of, and that's carbohydrates, lipids and proteins. So with the carbohydrates, there's actually more than one enzyme that's needed to hydrolyze them from the polysaccharides to the monosaccharides. We have amylases and membrane-bound disaccharidases. So digestion begins in the mouth, it will then continue in the duodenum, and then it's completed in the ileum. And the first thing is the amylase that is produced by the salivary glands is going to be doing the digestion in the mouth. But you also have amylase produced in the pancreas, and that is going to be releasing the amylase into the duodenum. And this hydrolyzes the polysaccharides into the disaccharide maltose, and that is by breaking the glycosidic bonds between the molecules in the polysaccharide. Then we have sucrase, and it could be lactase as well, which are membrane-bound enzymes that hydrolyze sucrose and lactose into monosaccharides. You could also have maltase, which would be hydrolyzing maltose into glucose. For proteins, these are large biological molecules, they're polymers, and they are hydrolyzed by 3 enzymes. We have the endopeptidases, which hydrolyze the peptide bonds between amino acids in the middle of a polymer chain. We have the exopeptidases, which are going to do hydrolysis and break the peptide bonds between amino acids at the end of the chains. And then the membrane-bound dipeptidases will be breaking the peptide bonds between 2 amino acids. So it hydrolyzes those dipeptides. Now, where this is actually happening is firstly in the stomach. So that's where it begins. It continues in the duodenum and then it's fully digested within the ileum. Lipids are digested by lipase or lipase and also through the action of bile salts. Now, lipase is produced in the pancreas and it can hydrolyze the ester bonds in triglycerides to form monoglycerides and fatty acids. What the bile salts do after they've been produced by the liver is emulsify lipids to form tiny droplets called micelles. And these increase the surface area available for the lipase to bind onto and therefore act on. So they're going to be making this digestion more efficient. So we can see here the physical and the chemical digestion. Physical is the emulsification and that micelle formation. Chemical digestion is the action of the lipase. So the physical, this is where the lipids are coated in bile salts to create an emulsion. Many of those small droplets of lipids provide a larger surface area to make hydrolysis faster. And as we said, the chemical is just the action of lipase. Now, a micelle is a vesicle formed of fatty acids, glycerol, monoglycerides and bile salts, but we're going to have a look at how that aids in the absorption of lipids. So lipids are digested into monoglycerides and fatty acids by the action of lipase and the bile salts. And that is how we create these tiny structures, which are the micelles. When the micelles encounter the ileum epithelial cells, due to that non-polar nature of the fatty acids and monoglycerides, they can simply diffuse across the cell surface membrane to enter the cells of the epithelium. Once in the cell, these will then be modified back into triglycerides inside of the endoplasmic reticulum and the Golgi body. And then they can form vesicles and be released from the cell into the lacteal and be transported around the body. So absorption in mammals is taking place in the ileum. And this is where it links to the villi and the microvilli and the adaptations that these structures have. So the ileum wall is covered in villi, and they have thin walls surrounded by a network of capillaries. And they also are covered in epithelial cells that have even smaller microvilli on them. And that is creating that large surface area. The fact that the walls are so thin is providing a short diffusion distance, and that network of capillaries is maintaining the concentration gradients. Now, this actually links to as well topic 2 when you learnt about co-transport. And that is because the monosaccharides like glucose and amino acids can only be absorbed by active transport in the form of co-transport. And that is because there's usually a higher concentration of either amino acids or glucose already in the epithelial cell. So in order to be able to move even more from the ileum into the epithelial cell, we require this co-transport. But that theory is covered in topic 2. Now, hemoglobin is involved in the mass transport of oxygen around the body. And it's an example of a quaternary structure protein because it's made up of 4 polypeptide chains. You also have a range of different types of haemoglobins. One that we'll be looking at is myoglobin, which is found in the muscle tissue in vertebrates and also in fetuses. The oxyhemoglobin dissociation curve is a way to look at how haemoglobin behaves in different conditions. Oxygen is loaded in regions with a high partial pressure of oxygen. So what that means is when haemoglobin is in areas with a lot of oxygen available, such as the alveoli, it will be able to pick up lots of oxygen. In regions with a low partial pressure of oxygen, for example respiring tissues, haemoglobin unloads the oxygen. And that is how we get the shape of this oxyhemoglobin dissociation curve. We can see at the different partial pressures how saturated haemoglobin is with oxygen. And the more saturated it is, that means it must have loaded up with more oxygen. The less saturated it is, it must have unloaded oxygen, and that's why it's not holding very much anymore. So we can see here we've got our respiring tissues compared to the alveoli. Now this graph also demonstrates cooperative binding. And this cooperative nature of oxygen binding to haemoglobin is due to the haemoglobin changing shape when the first oxygen binds. It then makes it easier for the subsequent oxygens to bind. Now you can also see dissociation curves to demonstrate the Bohr effect. And the Bohr effect is when a high carbon dioxide concentration or partial pressure causes the oxyhemoglobin curve to shift to the right. The affinity for oxygen decreases, and that is because the acidic carbon dioxide changes the shape of the haemoglobin slightly, and therefore it means the haemoglobin behaves differently and it's more likely to unload the haemoglobin even at the same partial pressures. So we can see here we've got three curves at different pHs, and the lower the pH, the more carbon dioxide there would be present. So we can see here at pH 7.6 compared to, let's say, 7.2, the curve has shifted to the right, and even at the same partial pressure, so we'll pick 20, we can see that the saturation is only about 25%, but for this one, it's just under 60%, and that is because of this Bohr effect. So low partial pressure of carbon dioxide would typically be in the alveoli because you are exhaling that carbon dioxide. A high partial pressure of carbon dioxide would be at respiring tissues because carbon dioxide is produced in respiration. Now that would be an advantage because it means that haemoglobin will behave differently, it will unload oxygen more readily, and therefore it's unloading the oxygen at the respiring tissues. Now, different animals have haemoglobin adapted to their particular needs and environments also. And that is one thing that you could get application questions on. So a fetus, and this is a human fetus, they will have myoglobin or fetal haemoglobin, and the fetal haemoglobin has an even higher affinity for oxygen even at the same partial pressures compared to adult haemoglobin. And that's an advantage because it means that as the blood is circulating through the umbilical cord, the fetus's haemoglobin is able to load the oxygen off of the mother's adult haemoglobin. Llamas are found at high altitudes where we have very, very low partial pressures of oxygen. So for the llama, we can see that they also have haemoglobin that has a higher affinity for oxygen, even at lower partial pressures. So we can see that would mean that if there isn't very much oxygen available, which there wouldn't be at a high altitude, then the haemoglobin is still able to load up. Animals like doves, for example, their needs are that they need more oxygen to match their faster metabolism because they're flying so much, they need oxygen for the muscle contraction. So the haemoglobin of a dove, the curve actually is shifted to the right compared to human haemoglobin. And that means that the haemoglobin has a lower affinity for oxygen, and therefore it will more readily unload the oxygen which is needed for respiration. Earthworms are a different example, so they will be underground a lot where there's very low partial pressures of oxygen. So they have hemoglobin that has very high affinities, even at low partial pressures, so that their hemoglobin can load up with whatever oxygen is available. In mammals, the circulatory system is described as closed and double. Closed meaning that the blood remains within the blood vessels the entire time, and double referring to the fact that the blood passes through the heart twice in each circuit. So there is one circuit that delivers blood from the heart to the lungs, and the other circuit delivers the blood from the heart to the rest of the body. Mammals are required to have a double circulatory system to manage the pressure of blood flow. The blood flows through the lungs at a lower pressure, and this is to prevent damage to the capillaries in the alveoli, but it also means that the blood will flow at a slower speed, so there is more time for gas exchange. The oxygenated blood from the lungs then goes back to the heart and it's pumped out at a higher pressure to the rest of the body. And this is important to make sure that the blood is able to reach all of those respiring cells in the body. Now, the key blood vessels that you need to know about are, first of all, the coronary arteries, and these are the arteries that cover the heart itself to supply the heart muscle or cardiac muscle with oxygenated blood. You also need to know the 4 blood vessels that are delivering blood into and out of the heart: the vena cava, aorta, pulmonary artery, and the pulmonary vein. You need to know the blood vessels that deliver blood to the lungs and carry it away, so the pulmonary artery and the pulmonary vein. So any time that you see pulmonary, that is referring to the lungs. And you can see here that the pulmonary artery is carrying blood away from the heart to the lungs. The lungs will then oxygenate the blood, and the pulmonary vein is delivering that blood back into the heart. The kidneys, we have the renal artery and the renal vein. So where you see the word renal, that is referring to a blood vessel attached to the kidneys. Those major blood vessels are connected within this double circulatory system via arteries, arterioles, capillaries, and veins. The cardiac muscle has a range of special features. Now, the walls of the heart have a very, very thick muscular layer so that it can contract with high force to deliver high pressure blood to all of the body cells. Now, the unique properties that the cardiac muscle has is, first of all, it's myogenic, and that means it can contract and relax without nervous or hormonal stimulation. It also never fatigues. So as long as it has a constant supply of oxygen and glucose, it will be able to respire aerobically. The coronary arteries we can see here, they are what supply the cardiac muscle with this oxygen and glucose so that it never fatigues. They branch off from the aorta, which we can see here. And if one of those coronary arteries was to become blocked, that would then mean that the heart muscle or the cardiac muscle wouldn't be receiving the oxygen or glucose, therefore the cardiac muscle wouldn't be able to respire and it would stop contracting. And that would cause a myocardial infarction, or in other words, a heart attack. So you need to know some of the key structures of the heart. First of all, there are 4 chambers. We have 2 atria at the top, and we have the 2 ventricles at the bottom. The atria have thinner muscular walls, and that's because they don't need to contract with as much force because they're only delivering the blood from the atria into the ventricles. They also have elastic walls so that they can stretch when the blood is entering. The ventricles have much thicker muscular walls, and that is so they can contract with more force and pump the blood out at higher pressure because they are carrying the blood further distances, either to the lungs or to the rest of the body. Now, the right ventricle is pumping the blood to the lungs, and that is at a lower pressure, as we said, to prevent damage to the capillaries and go at a slower speed. So, comparatively, the right ventricle wall has a thinner muscular layer. The left ventricle has a much thicker muscular layer in its wall because it has to contract with more force to pump the blood at high pressure around the body. Highlighted here we have the 4 key blood vessels. We've got the aorta and the pulmonary arteries, the vena cava and the pulmonary veins. But for some of them there's actually multiple. So we can see we have the superior vena cava and the inferior vena cava. We also have the pulmonary artery, which is carrying the blood away from the heart to the lungs, but we have one coming out of the left and one out of the right. Same with the pulmonary veins, which is carrying blood into the left atrium from the lungs. We have one on the left side, one on the right side. And the reason for both of those is we have a right lung and a left lung. The aorta is carrying blood from the left ventricle to the rest of the body. Now, some of the ways just to try and remember what each of these are doing are, first of all, if you see veins, veins are carrying blood into the heart. And vena cava means vein, and cava means body. So that is carrying the deoxygenated blood from the body into the right atrium. Pulmonary, we said, means lungs, and again it's a vein. So it's carrying oxygenated blood from the lungs into the left atrium. Arteries, think A away, it's carrying blood away from the heart. So a pulmonary artery is carrying deoxygenated blood from the right ventricle to the lungs. And the aorta is the major artery, which is carrying oxygenated blood from the left ventricle to the rest of the body. Now, the valves that you need to know about are the semilunar valves, which are in the aorta and the pulmonary artery, as well as the atrioventricular valves, which are between the atria and the ventricles. Sometimes you'll see those called the bicuspid and the tricuspid valves. And we can see these labelled here on the diagram of the heart. Now, valves are to prevent the backflow of blood, and they do this by only opening when the pressure is higher behind the valve compared to in front. So if the pressure is higher in front, it causes the valve to shut, and that is how it stops the blood from flowing backwards. The septum is running through the middle of the heart to separate the blood on the deoxygenated and the oxygenated side, and that's to help to maintain a high concentration of oxygen in the oxygenated blood to make sure that these diffusion gradients are maintained so that diffusion can occur at respiring cells. Looking at the blood vessels that are connecting all of the major blood vessels together, we have the arteries, arterioles, capillaries and veins. Arteries, as we said, think A away, they carry blood away from the heart towards the arterioles. The arterioles are smaller than arteries and connect to the capillaries. The capillaries connect the arterioles to the veins, and the veins will then carry blood back into the heart. So we can see here our arteries connected to our arterioles. We then have this capillary bed or network of capillaries connecting to the venules and then the vein. So the structures of the arteries and vein are quite different because of the fact that one is carrying blood away from the heart and one's carrying it into, so the pressure of the blood they're carrying will vary. So arteries have a much thicker muscular layer compared to the veins, and that is so that constriction and dilation can occur to control the volume of blood flowing through them. They also have a thicker elastic layer than the veins, and that's to help maintain the blood pressure. So the walls can actually stretch and recoil in response to the heartbeat. So overall, the thickness of the wall is thicker because of those two thick layers, but also it stops the blood vessels from bursting at that high pressure. So we can see here how thick those walls are. They also don't have any valves. So in comparison, veins have a much thinner muscular layer, thinner elastic layer and thinner walls in general, and that is because the blood is at lower pressure, so they're not at risk of bursting. However, because the blood is at lower pressure, they do require valves to help prevent the backflow of the blood, to make sure that the blood is going to be pumped back into the heart. The capillaries are very, very narrow in diameter, and this is to make sure that the blood speed is going to slow right down as it's passing through the capillaries. And that is to allow time for gas exchange and tissue fluid formation in the capillaries. So we've already talked about the arteries and the veins, but if we now add in the arterioles and capillaries, the arterioles are thicker than in the arteries, the muscular layer, and that is to help restrict the blood flow to the capillaries. The elastic layer is thinner than the arteries, and that is because the pressure is now slightly lower. And overall, the wall is thinner compared to the arteries because the pressure is slightly lower. But they still don't have valves. Capillaries do not have any muscular or elastic tissue layer. They are only one cell thick, and that is to make sure there's a really short diffusion distance, because the function of the capillaries is that is where exchange of materials between the blood and the cells occur. The cardiac cycle is split into 3 stages. Now people pronounce this differently, so I'm going to say both: diastole or dystole, atrial systole or atrial systole, and ventricular systole or systole. And we're going to go through what happens at each stage. So in diastole, the atria and ventricle muscles are relaxed. And this is when blood will enter the atria through the vena cava and the pulmonary vein. And the blood flowing into the atria then causes an increase in the pressure, because we've now got a larger volume of liquid there. Then we get atrial systole occurring, and that is when the atria muscles contract, and that increases the pressure even more in the atria. And because we now have this high pressure, that causes the atrioventricular valves to open, and blood moves from the atria into the ventricles. And at this stage, the ventricle muscles are relaxed. We have ventricular diastole. The last stage then we have is ventricular systole, which is when the ventricle muscles contract. And that happens after a short delay, and it will increase the pressure beyond that of the atria, which causes these atrioventricular valves to shut. But when they've contracted high enough to increase the pressure above that of the atria and pulmonary artery, the semilunar valves will open. And that causes blood to be pumped out of those 2 blood vessels. You could be asked to calculate the cardiac output, and that is the volume of blood which leaves one ventricle in 1 minute. And it can be calculated by doing the heart rate times the stroke volume. And the heart rate is beats of the heart in 1 minute. Stroke volume is the volume of blood that leaves the heart each beat, and that is in decimeters cubed. Tissue fluid is a fluid that contains water, glucose, amino acids, fatty acids, ions and oxygen, and it's the liquid that is forced out of the capillaries to bathe the cells, to make sure the cells are getting all of the minerals, nutrients that they require from the blood. Now it's formed due to the fact that capillaries have very, very small gaps between each of the cells that make up the capillary walls. And as the blood enters the capillaries from the arterioles, the smaller diameter results in a very, very high hydrostatic pressure of the blood. And because we have this high hydrostatic pressure and tiny gaps between the cells that make up the capillary walls, water and small molecules are forced out. So glucose, amino acids, fatty acids, ions, oxygen are all forced out of the capillary to the surrounding cells. And that is called ultrafiltration. So the molecules that can be forced out, we just went through all of those, but anything that is too big cannot get out of those tiny gaps between the capillary cells, and that would be things like the red blood cells, platelets, and large proteins. So those will remain in the blood. And the fact that they remain in the blood is actually what enables the liquid from the tissue fluid to be reabsorbed again. So towards the venial end of the capillary, which is over here, the hydrostatic pressure has decreased, and that's because so much liquid has been forced out. The pressure within the capillary has now dropped by the time we've got to the venial end of the capillary. However, we don't have as much liquid, but we have lots of those large proteins that have remained behind, which has lowered the water potential of the blood in the capillary compared to the water potential in the liquid surrounding the cells, which is the tissue fluid. And as a result, that liquid is going to, or the water is going to re-enter by osmosis, and it will carry with it waste that is dissolved in that tissue fluid that was released from these cells. So things like carbon dioxide and urea. So that gets absorbed into the capillaries by osmosis, and then it'll be transported around the body to be removed as waste. Now, not all of the liquid will be reabsorbed by osmosis, because eventually equilibrium will be reached and the water potential inside of the capillary and of the tissue fluid will be the same. So any of that water that doesn't get reabsorbed by osmosis will enter the lymphatic system instead, and then when we have it transported around the lymphatic system, it will eventually get to one of the lymph vessels near the heart where that liquid is drained into the blood again, and that is how we don't run out of liquid in the blood. So the last bit of topic 3 is mass transport in plants. And we start with the mass transport of water, looking at, first of all, transpiration. So transpiration is the loss of water vapour from the stomata. So water evaporates out of stomata on the leaves. And there are 4 key factors that affect the rate of transpiration. First of all, light intensity. The more light there is present, the more stomata will open, and therefore there's a larger surface area for evaporation. If it's hotter, that means that the water molecules will have more kinetic energy, and therefore they'll be moving faster and evaporate at a faster rate. With humidity, the more water vapour that is in the air, that will actually make the water potential more positive outside of the leaf, and it reduces the water potential gradient. So what that means is the more humid it is, the less transpiration there will be occurring. With wind, because the wind is carrying away air that contains water vapour, the wind will actually be maintaining the water potential gradient. So the more windy it is, the faster the rate of transpiration. So how water actually moves from the roots all the way up the xylem to transpire out of the stomata links to this idea of the cohesion tension theory. So water has to move up the plant from the roots against gravity, and that can be a very, very large distance if it's a huge tree. And this is possible because of the cohesion, the capillarity or adhesion, and root pressure. So if we look at cohesion first, this links to the theory of water. Water is dipolar, meaning we've got 2 slight charges, 2 different charges, a slight negative on the oxygen and a slight positive on the hydrogen atoms. And that means hydrogen bonds can form between the hydrogen and oxygen of different water molecules. And it creates this cohesion or sticking together of the water molecules. Now that's an advantage because as the water moves up the xylem, that means it's all bonded together with these hydrogen bonds, and it moves up as a continuous column of water, and it's much easier to pull up a column than it is individual water molecules. The next idea was this idea of adhesion, whereby the water molecules will actually also stick to the walls of the xylem. And if you have a really narrow xylem, that will increase this capillarity effect, and therefore the liquid will be moving up more just from this sticking, this adhesion effect. So the narrower the xylem, the easier it's going to be to transport the liquid up the xylem against gravity. Root pressure is the final concept, and this is as the water moves into the roots by osmosis, it increases the volume of liquid inside the roots, and therefore the pressure inside the root increases as well. And we call that root pressure. And that creates this positive pressure, which means it's a pushing force. Because we have lots of water in the roots, it pushes all of the water above it upwards, so it helps push the water up the xylem. So those 3 concepts all work together for the cohesion tension theory, and that's how water moves up against gravity. So just have a look at this as a whole. The first thing that happens is the water evaporates out of the stomata, and as that water vapour is leaving, that is then leaving behind this lower pressure because liquid has been lost. And that creates this negative pressure, or in other words, a pulling force. And that pulls the water column up the xylem. And because of cohesion, we have that water column whereby all the water molecules are stuck together. The water molecules are also adhering or sticking to the walls of the xylem to help pull the column upwards. And as the column of water is pulling up, It also creates tension in the xylem, which actually pulls the xylem inwards, making it narrower, which increases that impact of the capillarity or the adhesion, and it helps to move the water up even more. Now, the second type of mass transport in plants is looking at how the organic molecules like glucose produced in photosynthesis are transported around. And this now happens in the phloem. Now, the phloem tissue contains two key cells. We have the sieve tube elements and the companion cells. And the sieve tube elements, which are here, are living cells, but they don't contain any nuclei, and they're very few organelles. And that is so it's pretty much hollow to make it easy for the solutions to transport through the tube. The companion cells are on the outside, and they provide the ATP required for active transport of the organic substances. So they have all the organelles that the sieve tube elements don't, so they can provide the resources needed. Now, the transport of these organic molecules within solution is often explained using this source to sink model. So we have here the xylem next to the phloem, and the source in this model would be a photosynthesizing cell. The sink, which is where the sugars are going to be delivered, is a respiring cell. So at the source, we have photosynthesizing cells, and the sucrose or glucose that will be made in photosynthesis is going to lower the water potential of those cells. And therefore, any surrounding water in the plant or from the xylem is going to be entering those cells by osmosis. At the other end, where we have our respiring sink cells, because they'll be using up those sugars in respiration, there will be a more positive water potential inside of the cell compared to outside. And therefore, water is going to leave the respiring cells by osmosis to other cells in the plant or even the xylem. So the effect that has is there'll be an increase in the hydrostatic pressure in the source cell, but a decrease in the sink cell. And because of those pressure changes, the liquids that are within that source cell will be forced by that high hydrostatic pressure through the xylem all the way to the sink cell. Now, that is part of the story, but also you need to know the translocation steps. So how those sugars within the leaf cell actually make it into the phloem for that pressure then to actually move the liquid along. So photosynthesis is occurring in the chloroplasts, in the leaves, and we're calling those the source cells. That, we said, creates a high concentration of sucrose. Now, as well as that affecting the water potential, that also means that the sucrose can diffuse down its concentration gradient into the companion cell by facilitated diffusion. We then get active transport of protons or hydrogen ions from the companion cell into the space within the cell walls, and that uses energy because it's active transport. Now that creates a concentration gradient, and therefore the protons move down their gradient via carrier proteins into the sieve tube elements. And the co-transport of sucrose with those hydrogen ions occurs via a protein co-transporter, and that is how the sucrose goes from being in the companion cell into the phloem, even though there is a high concentration often of sugar or sucrose in the phloem already. So the next step then is we're looking at the movement of that sucrose within the phloem, sieve tube element. So the increase of sucrose in the sieve tube element is going to lower that water potential, and therefore water enters those sieve tube elements from the xylem vessels, which are tightly compacted next to the phloem. The increase in water volume in the sieve tube element at this position will increase the hydrostatic pressure, and this is where we have the idea of source to sink. It causes the liquid to be forced from the source area down to the sink cells. Lastly, the sucrose is then used in respiration at the sink, or it might be stored away as starch if it's not currently required. But that means more sucrose is actively transported into the sink cells, and that is going to cause the water potential to decrease. And as a result, will have water moving by osmosis from the sieve tube elements into the sink cell. Some water will also be returning from the sieve tube element into the xylem. The removal of that water is decreasing the volume in the sieve tube element, and therefore the hydrostatic pressure decreases. So the movement of soluble organic substances is due to the difference in the hydrostatic pressure between the source and the sink end of the sieve tube element. Now you could be asked about two particular investigations that prove translocation. The first one is called tracers, and this is where we have tracing involving radioactively labelling carbon. Plants are provided with only radioactively labelled carbon dioxide, and over time they'll be absorbing that in through the stomata, using it in photosynthesis, and the organic substances like the sugars created will all contain that radioactively labelled carbon. Thin slices from the stems are then cut and placed on X-ray film that will turn black when exposed to radioactive material. When the stems are placed on the X-ray film, the section of the stem containing the sugars turn black, and this highlights where the phloem are, and it can show the sugars are transported in the phloem, and it also means you can track the route that is taken. Ringing experiments is when a ring of bark and phloem are peeled and removed off the trunk, like we can see here. The result of removing the phloem is that the trunk swells above the removed section, and analysis of the liquid in this swelling shows it contains sugar. So this shows that when the phloem is removed, the sugars cannot be transported, and it therefore proves the phloem transports sugars. So we start topic 4 by comparing the DNA in eukaryotic cells and prokaryotic cells. And comparisons mean similarities and differences. So our two key similarities are the fact that the DNA is made up of DNA nucleotides for both, meaning it contains deoxyribose, a phosphate group, and a nitrogenous base. The nucleotides are also joined together by phosphodiester bonds to make the polymer chain. The three key differences are that eukaryotic DNA is longer, eukaryotic DNA is linear, so it occurs as straight lines in the chromosomes, whereas prokaryotic DNA you get circular loops of DNA. The DNA in eukaryotes is associated with histones, but for prokaryotic DNA it is not associated with proteins, those histones. So those are your key differences in the DNA. And this is just showing you here the organisation of the DNA in a eukaryotic cell. So we have our chromosomes within the nucleus that is tightly coiled to make the chromosome. But if we unwind that, you can then see the nucleosomes, which is actually the DNA wrapped around the histone proteins, and that is how we tightly coil to fit it all in the nucleus. Now you also need to be able to compare the DNA that is found in the mitochondria and chloroplasts of cells to the DNA that you find in prokaryotic cells, because they actually have quite a few similarities. So we can see here our mitochondria, and you have just a loop of DNA, and the same within your chloroplast. You just have this circular loop of DNA, and they have their own DNA so that they can translate their enzymes that they need for photosynthesis in the chloroplast and respiration in the mitochondria. So the similarities are they are both short sequences of DNA, they are both circular, and neither of them are wrapped around histone proteins. So that then takes us on to looking at what a gene is. And a gene is a sequence of DNA, and it codes for the amino acid sequence for a particular polypeptide and also a functional RNA. So it codes for an mRNA molecule. And we can see here one of the extra key terms as well, and that is locus. And locus is the exact position that one particular gene is found on a chromosome. So locus is location, so that's the way to remember it. Locus, location of the gene. Okay, so the genetic code, you need to know a few features of the genetic code. One thing, first of all, is knowing that a sequence of 3 bases on DNA is called a triplet, and those 3 bases will code for a particular amino acid. So the 3 features are: it is a degenerate code, it's universal, and it's non-overlapping. So if we have a think about this idea of degenerates, there are 20 amino acids that we said exist, and we have 4 possible DNA bases. And the way they actually worked out that it was 3 DNA bases that codes for 1 amino acid was actually mathematically. They looked at all the possible code options you could get if you only had 1 base coding for 1 amino acid, and that would only give you 4 possible codes, G, C, T or A. That's not enough to code for 20 different amino acids. So then if you think about, could it be 2 bases? If you had only 2 bases, that would give you 16 possible different codes, and that's still insufficient. So they realised it must be 3 bases that code for 1 amino acid, because that actually gives you 64 possible different options for what those triplets of bases could be. And that is more than enough to code for the 20 amino acids. And that is why the genetic code is degenerate. And what we literally mean by this definition is there are more than 1 triplet of bases that codes for the same amino acid. That would be your key definition. And this table here is just showing you an example of that. So you can have a look, for example, at, let's look at glycine here, GLY, GGG, GGA, GGC, GGU. All 4 of those triplets of bases code for glycine. And this is an advantage of the genetic code, because if there is a mutation and one of the bases in a triplet is changed, you might still have the new triplet coding for the same amino acid, and therefore it has no effect on the overall polypeptide chain. Universal means that the same triplet of bases codes for the same amino acid in all organisms. Non-overlapping is the fact that each base is only involved in 1 triplet. So if we just draw boxes around this to show you what I mean, this base A is only in this 1 triplet, and this C is only in the triplet, this G is only in this triplet. We don't have this G also making up a second triplet of bases. So every codon or triplet of bases is read as a discrete unit. This is an advantage, as if a point mutation occurs, it will only affect 1 codon and therefore 1 amino acid, so it will minimize any potential harm. Now in your DNA, you have sections of base sequences, which are introns, and you have sequences of DNA that are exons. Introns are sequences of DNA bases that do not code for polypeptides. And you actually have a lot of introns making up your nuclear DNA. The exons are sequences of DNA bases that do code for the amino acids. So the exons are the coding regions. And when we say codon, a codon is 3 bases on mRNA that codes for a specific amino acid. A start codon is 3 bases that you find at the start of every gene, and that is what initiates translation to occur. A stop codon is the final 3 bases that you have at the end of every gene. And those 3 bases will cause the ribosome to detach during translation, and therefore it stops the translation of the polypeptide chain. A genome is what we call an organism's complete set of genes in a cell. So that is your definition of a genome, whereas the proteome is the full range of proteins that a cell is able to produce. The genome should never change unless there are mutations, whereas the proteome of a cell can constantly change depending on which proteins are needed in a specialized cell, because you'll have some genes have been switched off or on, and that's what makes it specialized. The genome of an organism will really differ between different species. So for example, a bacteria contain on average 600,000 DNA base pairs within their genome, whereas humans, we have 3 billion DNA base pairs. So this then starts to take us on to RNA before we get into protein synthesis. So messenger RNA is what mRNA stands for, and we can see it here on the picture. It is short compared to DNA because it's only a copy of one gene, whereas the DNA is the entire genome. It's single-stranded and it's found in both the cytoplasm and the nucleus. So it is made during transcription, and that happens in the nucleus. But then once it's been modified, it leaves the nucleus, enters the cytoplasm to attach to a ribosome. It's 3 bases on mRNA that are codons, so 3 bases which can code for a particular amino acid. tRNA is transfer RNA, and this is found in the cytoplasm. It has an amino acid binding site, which we can see up here at the top, and each tRNA molecule will have a particular or specific amino acid attached to that binding site. The tRNA molecule also has 3 bases on it at the bottom here, and we call those 3 bases the anticodon, and they will be complementary to a particular codon on mRNA. And when those align, they're held in place so that amino acids can start to bond together during translation. So tRNA is involved in translation, the second stage of protein synthesis. A ribosome will be holding it in place to enable the joining of amino acids. It has this clover leaf shape, that's what we call this shape, and we can see here these lines are representing hydrogen bonds. So it's still single-stranded, it's just folded to create this shape, and that shape is held in place by hydrogen bonds. So that then takes us on to protein synthesis, and it's split into 2 steps: transcription, which is where one gene at a time from DNA is copied into mRNA. Then we have translation, where the mRNA will join with a ribosome, and corresponding tRNA molecules will then bring specific amino acids. So first of all, we have transcription. So a complementary mRNA copy of one gene on the DNA is created in the nucleus. mRNA is much shorter, we've already said, and that is because it's only copying one particular gene, and therefore it's able to leave the nucleus because it is smaller. Your key steps are all here. So if you did have a long answer question, describe the process of transcription, these would be your key marking points, with the key marks put in bold. So first of all, the DNA helix unwinds to expose bases, and you have one strand acting as a template, and that's our second mark. Like with DNA replication, that is caused by DNA helicase breaking the hydrogen bonds. In the nucleus, you then have free-floating mRNA nucleotides, and they will align opposite their complementary DNA base pairs on the template strand. The enzyme RNA polymerase will then join together those RNA nucleotides to create the mRNA polymer chain. Once it's copied, it then has to be modified, and then it leaves the nucleus via a nuclear pore. The modification that happens is this here, splicing. In eukaryotes, after transcription, we actually call the molecule pre-mRNA, and that is because it still contains the introns, which are those non-coding sequences of bases. And that's because the DNA, the gene that was copied, there will be introns within it, which are the non-coding sequences. So the RNA that is copied will still contain those introns. So the introns need to be removed, and we call that splicing. They're spliced out, so cut out, they're spliced out by a protein called a spliceosome. And now we have finished mRNA that is ready to leave the nucleus. That stage doesn't happen in prokaryotes because they don't have introns. Translation is the next stage in the creation of the polypeptide chain, and it involves both mRNA and tRNA. If you were asked to describe that whole process, again, these are your 6 key marking points, and in bold are the key terms you would have to include. Once the modified mRNA has left the nucleus, it will then bind to or bind with a ribosome in the cytoplasm. The ribosome will attach at the start codon of the mRNA molecule. The tRNA molecules with complementary anticodons to the start codon will then align opposite, and they're held in place by the ribosome, which we can see here in the picture. The ribosome holds together 2 tRNA molecules at a time. The 2 amino acids that have been delivered by the tRNA molecule are joined by a peptide bond. And that reaction does require energy in the form of ATP and an enzyme, but you don't need to know the name of it. Once that happens, the tRNA molecule will be released and the ribosome moves along one codon, so the next tRNA molecule can then align its anticodons to its codons. So this continues until the ribosome reaches the stop codon at the end of the mRNA molecule, and when it does, that causes the ribosome to detach and therefore translation ends. Now the modifications, because we now just have a polypeptide chain, the modifications will occur in the Golgi body for folding to create that secondary, tertiary or quaternary structure. We then move on to how variation is introduced, and gene mutations is one way. A change in the base sequence of DNA is what a gene mutation is, and they randomly occur during DNA replication, so within the interphase part of the cell cycle. These random mutations are more likely to occur if you're exposed to mutagenic agents, which can interfere with the DNA replication. That includes high energy radiation like UV light, ionizing radiation like gamma rays and X-rays, and also some chemicals which we call carcinogens, for example mustard gas and cigarette smoke. A gene mutation can result in either a base being deleted or swapped, so substituted for a different base. So here are our examples. We have our original DNA sequence. This is showing a substitution. Instead of cytosine, that has been swapped or substituted for adenine. This one is showing a deletion because that base C has now been deleted. And that's actually caused what we call a frame shift. Everything downstream of the mutation has shifted to the left. A base mutation, though, might have no impact at all because the new codon may still code for the same amino acid, and that's because the genetic code is degenerate. Chromosome mutations can also occur, and chromosome mutations are changes in the number of chromosomes, and this spontaneously occurs during meiosis in a process called non-disjunction. So non-disjunction is when the chromosomes, or it could be the chromatids, do not equally split during anaphase of either meiosis 1 or meiosis 2. So that's what we can see here, non-disjunction occurring because the chromatids didn't separate and instead all of them are being pulled to the same pole of the cell. Now this can occur in two forms. Either a change in the whole set of chromosomes, which we call polyploidy, or changes in the number of individual chromosomes, which is aneuploidy. So we'll go through polyploidy first, which we said is a change in the whole sets of chromosomes. So you could end up, instead of being diploid, having 2 copies of every chromosome, which we have in humans, you could have 3 copies or 4 copies of every chromosome, which would be called triploid or tetraploid. Now in humans that would be fatal. You don't see triploid or tetraploid humans, but it's actually quite common in plants. So how this would occur then? Each homologous pair is doubled in replication, and that happens in interphase. In this example, we have non-disjunction in meiosis 1. For some reason, the spindle fibers haven't attached to the chromosome on this side, and they have attached to all of the chromosomes on the other side. So when the spindle fibers retract, it's going to pull all of the chromosomes to one side of the cell, and therefore they're all going to be in this cell and there'll be no chromosomes in the next one. In meiosis 2, that would mean that these 2 gametes will contain no chromosomes at all. So those gametes will not function. These gametes, though, meiosis 2 is happening normally, and we do have complete separation of all of the chromatids, but we now have 2 copies of every chromosome in the gamete. So instead of having a haploid gamete, we have a diploid gamete. And if a diploid gamete fuses with a haploid gamete, that is how we then end up with 3 copies. So we get 2 chromosomes from this gamete instead of 1, and we get just 1 chromosome from the haploid gamete. So that is polyploidy, changes in the whole sets of chromosomes. That could also happen if you have non-disjunction in meiosis 2. In this example, we can see the chromosomes in meiosis 1 did separate equally. And then we had normal meiosis 2 in this example, so we have 2 haploid gametes. But for this cell, in meiosis 2, there was non-disjunction. So the spindle fibres didn't form on this side, so the chromatids aren't separated equally, and instead they're all pulled to this cell. So again, we end up with a 2N gamete, a diploid gamete, and this gamete has no chromosomes in. Aneuploidy, this is different. This is when you have changes in the number of individual chromosomes. So sometimes individual homologous pairs of chromosomes fail to separate during meiosis. It's still called non-disjunction, but instead of it being affecting every single chromosome or chromatid, it's just one. And this is how Down syndrome occurs. You have non-disjunction on chromosome 21, so you end up with 3 copies of that chromosome instead of 2. So let's see how that might occur. We can see in this one, we have non-disjunction occurring at meiosis 1 because these spindle fibres for just that one chromosome or that one homologous pair of chromosomes is attaching and it pulls them both to this cell. And this cell does not get a copy of that red chromosome. If meiosis 2 occurs normally, so no non-disjunction, all of the chromatids are separated equally. However, because of the non-disjunction in meiosis 1, this gamete is haploid. It has 1 copy of all the chromosomes except for the red. So we describe that as haploid plus 1 extra chromosome. So N plus 1. These 2 are still haploid, but they're missing a chromosome. So we describe it as N for haploid minus 1. Now if an N plus 1, so a haploid with an additional chromosome, is to fuse with a haploid chromosome, that is how you can get trisomy, which means 3 copies, so trisomy is 3 copies of one particular chromosome. And that is how Down syndrome occurs, 3 copies of chromosome 21. Now you could also have non-disjunction occurring in meiosis 2. So we can see the normal cell division occurred in the first round of meiosis, but now we have non-disjunction in meiosis 2 because the chromatids are not separated equally for the red chromosome. They're all pulled to this one gamete, so that would be N plus 1. And this one is missing the red chromosome, so it's N minus 1. Now another way that variation can be introduced is in meiosis. And meiosis creates gametes, and it creates 4 genetically different haploid gametes by 2 nuclear divisions. So meiosis is how variation can be introduced as well, and that's through 2 mechanisms: independent segregation of homologous chromosomes and crossing over between homologous chromosomes. And both of these occur within the first round of division in meiosis. Independent segregation is when the homologous pairs of chromosomes line up opposite each other at the equator to form bivalents. It is random which side of the equator the paternal and maternal chromosomes from each homologous pair align. So we can see on this side by chance 2 purples, 2 reds, but equally it could have been a purple and a red and a purple. In meiosis, those homologous pairs of chromosomes are separated in meiosis 1. So one of each homologous pair ends up in the daughter cells eventually. This creates a large number of possible combinations of chromosomes in the daughter cells produced. And you can actually calculate this by doing 2 to the power of n. 2 because it is homologous pairs, so you have pairs of chromosomes, and n you would substitute in for how many homologous pairs of chromosomes that species has. So for humans, that would be 2 to the power of 23. We have 23 pairs of chromosomes, which means we can make over 8 million different possible gametes just from independent segregation. Now, crossing over also occurs sometimes. It's actually quite rare, but it can occur. And again, it occurs when the homologous pairs of chromosomes line up at the equator and form a bivalent, which is what we call it when you have both of them next to each other. And you have chromatids from each of those chromosomes cross over and they can get twisted around each other. That puts tension on the chromatids, causing part of the chromatid to break and swap. And in doing that, we create new combinations of alleles, which is represented by the letters here. So originally, this chromosome only had a dominant allele, but now we've got a dominant and recessive. So when that's pulled apart, we've got new combinations of alleles on that chromosome. Now, comparing meiosis and mitosis, meiosis is 2 nuclear divisions, whereas mitosis is only 1. That's why meiosis results in haploid cells, whereas mitosis is diploid cells. Meiosis introduces genetic variation through crossing over and independent segregation. Mitosis creates genetically identical cells. Now, you could be asked to identify meiosis in an unfamiliar life cycle. And what you need to do here is look for where you have cells that were diploid or 2N dividing to then create cells that are haploid, because that's what happens in meiosis. You go from 2N to N. It won't always be gametes because not all organisms have life cycles like humans where it is the creation of the gametes that is meiosis. So for example, we can see here we have the zygote, which is 2N, and then it makes something called zoospores, which are N. So that would be the meiosis stage. So that's what you're looking for, 2N moving to N. So again, that bit there would be meiosis. Genetic diversity, this is the number of different alleles of genes in a population. And this is what enables natural selection. Natural selection can only occur if there is genetic diversity. Natural selection is the process that leads to evolution. And our definition of evolution is the change in allele frequency over many generations. Natural selection is really important to the survival of the whole species because it results in the species becoming better adapted to their environment. And these adaptations might be anatomical, physiological or behavioural. So the key marking points that you'd need to describe for this process are, first of all, you would have new alleles for a gene being created by random mutations. If those new alleles increase the chances of the individual surviving in that particular environment, then they're more likely to survive and therefore more likely to reproduce and pass on that new advantageous allele to the next generation. Over many generations of that occurring, that new allele will now become more common in the gene pool, or in other words, we've increased the allele frequency. Now, the types of selection that occur are directional selection and stabilizing. Directional selection is when the advantageous allele is coding for an extreme trait. So this links to your antibiotic resistance example. And if we think about the traits being low resistance, medium, high resistance, the extreme traits would be either very low resistance or very high resistance. And in the case of antibiotic resistance, when there was a change in the environment, which is introduction of antibiotics, the bacteria which had the alleles for high resistance to antibiotics were more likely to survive and pass on that allele for antibiotic resistance. And that's why we saw this shift and antibiotic resistance allele became far more common in the species. Stabilizing selection is when whatever is the middling trait remains the selective advantage. And that would be the case if there's no change in the environment. And this is exemplified by human birth weights. So being a middling birth weight increases your likelihood of surviving, because if you're very, very small, then you might be very premature, might have underdeveloped lungs, difficulties regulating your temperature, and more vulnerable to infection. If you're very, very heavy, then it's going to be a more complicated birth, and that could result in difficulties. So that would mean that the modal trait remains a selective advantage, and we can see that the range of traits or alleles decreases over many, many generations. The range and the standard deviation decrease. Next then, it's thinking about species and taxonomy. So a species is when two organisms are able to produce fertile offspring. And a species must reproduce and pass on advantageous alleles in order for the whole species to be able to survive. And this is where courtship behaviour comes in. This behaviour is essential for successful mating, meaning mating and creating fertile offspring, in order for species to survive. So courtship behaviour is a sequence of actions which is unique to every species. So it is genetically coded for, this behaviour, and it is how animals are able to identify members of their own species to make sure they are reproducing with members of their own species to make sure that they can create fertile offspring. The behaviour, the sequence of actions, is normally carried out by the male, and then the female picks whether they are worthy of mating with. Now, this sequence could include dance moves, creation of sounds, release of pheromones, display of feathers, fighting. Whatever it is always a unique sequence to that species. The female then observes the ritual and decides if they look like they have a good enough set of alleles based on their fitness, their performance, to mate with. So the reason this is important is, we said, to ensure successful reproduction. And the way it does that is, first of all, because the behaviour is unique to every species, it allows them to recognise members of their own species and also the opposite sex. That is because to make fertile offspring, you'd need sperm and egg to fuse. It also synchronises mating behaviour. And what this means is it makes sure that the male and the female are mating when they are sexually mature. So the female is releasing eggs and the male is able to produce and release sperm. It can actually also help the survival of the offspring once it's born in some animals. And that's because this behaviour, this courtship ritual, can help form a really strong bond, which we call a pair bond, between the parents. So they're more likely to stay together. And if they are together, for some animals, like penguins, it increases their likelihood of survival, because you need one penguin to look after the chick, one penguin to go and find food. It also enables that strong and healthy mates are selected for, and therefore the advantageous alleles are being passed on to the next generations to ensure the survival of the entire species. So this could be used as a way for us to identify how closely related different species are as well. So for example, we've been shown 3 ducks here and the sequence of their courtship behaviour. And we can see that ducks 1 and 2 must be more closely related because their sequence of behaviours in the courtship ritual are more similar than duck 1 and 3 or ducks 2 and 3. And because this behaviour is genetically coded for, because the sequence of behaviours is more similar, their DNA-based sequence is likely to be more similar too. Phylogenetic classification is another way to look at how closely related different species are, and also how recent their shared common ancestors were. Phylogenetic classification is arranging groups according to evolutionary origins and relationships. So humans and chimpanzees, we can see, are most closely related to each other because they branch in the tree most recently compared to the others. So that means they evolved from a shared common ancestor more recently than they did compared to any of the other species. So that means they have had less time to accumulate different mutations in the human and chimpanzee populations compared to, let's say, the human and the horse population, because humans and horses, their recent common ancestor is back here. And there's no time scale on this, but this is normally going back, like this would probably be at the very start here. This could be looking at maybe 13, 20 million years ago. and so on. So it is millions of years of accumulating mutations in these separate species. Now you can also classify and group using a hierarchy. And a hierarchy is when you have smaller groups arranged within larger groups, and there's no overlap between those groups. So this is one particular hierarchy you need to know off by heart. Domain, kingdom, phylum, class, order, family, genus and species. And we can see here that within one genus you can have multiple different species. So that is our smaller groups within larger groups. But there's no overlap in those species. They're still distinct groups. The binomial system is a universal way of identifying organisms, and it's using two names. That's what binomial means. The first name is the genus, and the second name is the species. So for humans, our genus is Homo, and our species is Sapiens. So our binomial name is Homo sapiens. And we can see an example here of common names, which would be unique to every language, versus the binomial system, which is universally used. That gives you more information on how closely related they are, because calling them both robins is misleading, that they are closely related. But actually, we can see they're not the same species, and they're not even the same genus. So the final thing is looking at biodiversity. And there's different ways that you can classify and measure biodiversity. It could be looking at the range of different habitats that exist. It could be the variety of genes amongst all the individuals of a population of one species. Or it could be looking at the number of different species and individuals within each species in a community. Species diversity as well, if we go into that further, that takes into account species richness. And species richness just means the number of different species in a community. Biodiversity can be used to describe a range of habitats. You could be looking at the biodiversity of a very small local habitat like a forest, or it could be talking about the entire Earth. Having a low biodiversity isn't actually a cause for concern. For example, in the Arctic or deserts, you would expect that to be low. But if you have a decrease in biodiversity, that is a cause for concern, because that could be caused by human activity causing destruction. And farming techniques is an example of that. Farming techniques can reduce biodiversity, but it is needed to provide food for humans. So we have to come up with some kind of compromise between the two. You could get application questions where you have to suggest different farming techniques that could reduce the biodiversity. So I've got some of those listed here. And if you want more detail on that, then go to my actual biodiversity video to hear all of the examples and how it reduces the biodiversity and what the compromise would be. The way that we measure biodiversity is using the index of diversity. And that describes the relationship between the number of species in a community, which is the species richness, and the number of individuals within each species. So this is the formula to calculate it, and they would give you that in the exam. But they won't give you this. That capital N is the total number of organisms of all species. Lowercase n is the population size of one species. 1 is the lowest value you can get, and the larger the value, the greater that index of diversity, the greater the biodiversity. So here's an example. If you want to have a go, pause the video now and write it down and then have a look at the answer. If not, let's go through. So the first bit of the formula we're going to look at is the lowercase n times n minus 1. So that would be 6 as our lowercase, the population size of species A. And n minus 1, that would be 5. 6 times 5 would be 30. So we then do that for all of the others. And we need the sum of that. So the sum of that column is 180. Capital N, we said, was the total number of all individuals. And in this example, it's 25. So 25 times 24 divided by 180 is 3.3. Genetic diversity within or between species can also be measured by comparing different factors. So you can compare observable characteristics, but that can actually be quite inaccurate because members of different species that aren't even closely related might look similar because they live in similar environments. So more accurate ways to compare how closely related species are is through comparing the DNA-based sequence, the mRNA-based sequence, or the amino acid sequence for proteins. And the more similar those sequences, the more closely related they must be. So that is it for topic 4. I hope you found it helpful, and if you have, please give this video a thumbs up.